So welcome everyone. Good morning, good afternoon, good evening, whatever time zone you're in. We have people participating from all over the globe today. So my name is Nicole Kleinsteuer. I'm the director of the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICETM. And I have the privilege of being one of the co-chairs of the Microphysiological Systems for COVID Research, or the MPS Core Working Group. So along with my co-chair, Anthony Holmes from the UK's NC3Rs, and our other board members, we are delighted to welcome you to the spring virtual workshop of the MPS Core Working Group. And this workshop is really, I think it represents an evolution in this working group. We actually started planning this before we knew that COVID was going to be declared no longer a public health emergency of international concern. But coincident with the lifting of that status, we have also decided to investigate the broadening the utility of this working group beyond simply COVID research, and looking at the advancing research and regulatory acceptance of MPS for other infectious disease applications. So certainly still looking at COVID research, but looking a little bit broader in the infectious disease space. So very excited about the next 2 days. Next slide, please, John. Just a little bit of a refresher and background about the MPS Core Work Group. I think that many of you are already part of this work group, but for those of you who are not, we are always welcoming new members. So you can email Amber at the email address that's on the screen there. But the objectives of the MPS Core Work Group are really to provide this forum and facilitate engagement and collaboration and dialogue among international research and regulatory efforts in the MPS space. The beginning of the pandemic really signaled to us, obviously a grave public health emergency, but also an extraordinary opportunity in the field of MPS, given that many of the animal models of disease were not really translatable to the human scenario, and MPS were really coming to the front and center as providing really important information in the space of understanding the mechanisms of the disease, pathophysiology, testing new therapeutics, et cetera. So here we're really trying to make those connections and provide this space for researchers, regulators, and other investigators to interact with one another and get up to speed on the latest developments in the field. Okay, next slide, please. I've already mentioned my co-chair, Anthony Holmes. He's online. He'll be chairing tomorrow. And then our other board members from various NIH institutes. So Dan Tagley from NCATS, Candace Kerr and Mark Williams from NIAID, Kyle Glover and Tyler Goralski from Department of Defense, and then our incredible support staff from the NICETM group. None of this would be possible without these folks. So thank you so much for your assistance in organizing this meeting. Next slide, please. So just to give you a preview of the exciting day that we have in store for us. So we're going to kick things off with one of our Japanese colleagues discussing work in Japan, and then we're going to go through a really phenomenal morning or afternoon, wherever you are, of presentations. And then at the end, we're going to have a panel discussion, and we have some really exciting questions lined up for our panelists, and the panelists will be all of the speakers that you see on the agenda here today. Okay, so next slide, please. I'm sure that this is your first Zoom meeting and that you know nothing about these housekeeping basics. Just kidding. Just in case you need a refresher, you can, you have the controls at the top or bottom of your screen, depending on your version of Zoom. If you'd like to rename yourself so that we know what your affiliation is, that would be really helpful. So you can do that within the participants window. Next slide, please. And then as far as questions are concerned, you can put your questions in the chat. Please indicate which speaker your question is directed towards. We will, hopefully as time allows, we'll be taking clarifying questions at the end of every presentation. But as I mentioned, we have about an hour at the end for a panel discussion. And so if there's open-ended questions, perhaps those would be more appropriate for the panel. Feel free to put those in the chat as the day progresses, but maybe indicate that those are more suited for a panel discussion. And if you'd like to, we intentionally organized this as a Zoom meeting rather than a one-way Zoom webinar because we want to encourage dialogue and participation. So if you'd like to speak up verbally, you can do that by raising your hand, and then one of us will call on you and allow you to unmute yourself. Okay, next slide, please. And this workshop is being recorded for folks that were unable to join us today. So we had almost 170 people registered at last count. And so we expect to have lots of interest in the recording. And so we're going to make that available via the NICEDOM website. Okay, that's it from me. And I'm now delighted to turn it over to our colleague, Professor Seiichi Shida from Sojo University, who is kindly joining us despite the late hour in Japan. And will be speaking to us on the initiatives of the AMED MPS program in Japan towards regulatory acceptance of MPS data. Professor Ishida, over to you.
Okay, so thank you very much. Can you hear me and can you see me?
Yes, we hear you and see you very well.
Okay, so it was a great pleasure for me to have this opportunity to present our recent progress. Title of today's talk is Initiative of AMED MPS Project in Japan towards regulatory acceptance of MPS data. My name is Seiichi Shida from National Institute of Health Sciences and Sojo University. Today, I'm going to report an overview of the progress of MPS project in Japan. Next, please. So let me introduce myself briefly. I used to be the section chief of Division of Pharmacology in National Institute of Health Sciences, and currently I continue my research at Sojo University in the south part of Japan island, Kumamoto. I'm also continue to study in NIHS as a guest researcher. Next, please. My research interest is to reconstitute hepatic sinusoid in vitro and construct drug efficacy and safety evaluation systems. To do so, we are developing a culture system for hepatic stellate cells and establish a pathological model of hepatitis and liver fibrosis. And we are establishing a culture system capable of evaluating bioexcretion. For this purpose, we are using MPS to reconstitute the liver environment. First of all, I have to apologize that my research is not focusing to lung or COVID study. Next, please. Next, please. Yeah, okay. Today, I'd like to introduce the initiative of AMED MPS project in Japan toward regulatory acceptance of MPS data. AMED stands for Japan Agency for Medical Research and Development and is one of the largest funding agencies in Japan. There are 3 projects, MPS 1, which was finished last year, and MPS 2 and MPS RS, both are ongoing now. These 3 are major MPS projects in Japan. I'm going to explain what we did and what's going on now. Next, please. First is MPS 1 project. Next, please. This is the chart which shows how new test methods reaches the regulatory acceptance adopted from OECD's document. When the idea of a new test method comes to a researcher's mind, he or she begins research and development to turn the idea into a realistic testing method. AMED MPS 1 was responsible for this stage. AMED MPS 1 was supported by Ministry of Economy, Trade, and Industry. Next slide, please. This slide shows the organization of the AMED MPS 1. This project was launched in 2017 and finished last year. The annual budget was about $9 million. The project consists of 4 programs. Program 2 was developing and improving MPS device, and Program 4 was establishing the supply of hepatocytes from Japanese population. Program 1 was responsible for the production of MPS devices developed by Program 2 for commercialization. Program 3 standardized the MPS with cells provided by Program 1 and 2. Characteristics of this project was that pharmaceutical companies and device supply companies were also participating for developing models and conducting standardization tests for MPS. The project had central research center and open laboratory. Although not shown in the diagram, Pharmaceuticals and Medical Device Agency, PMDA, which undertakes the review of pharmaceuticals in Japan, participated in the project as an observer and exchanged opinion regularly. Next, please. As a result of 5-year dedication of participants, 4 MPS are close to the market. Top one is the device to evaluate the first-pass effect of the absorption in the small intestine and the metabolism in the liver. Second one can apply shear stress by perfusing of culture medium in the upper and lower side of a membrane. Third and fourth devices is to verify the interaction between 2 to 4 organs by connecting culture chambers. Next, please. The researcher participating in AMED-MPS-1 project developed the human iPS cell-derived small intestinal epithelial-like cells called FI-seq. FI-seq shows similar characteristics as in vivo human small intestine. These graphs show expression of drug metabolism-relating enzymes, transporters, and small intestinal epithelial cell markers. Dark gray bars are the expression data of human small intestine sample, and red bars are those of FI-seq. As you see, most of them are close to human small intestine sample. Light gray bar are those from Keiko2 cells. FI-seq is now available from Fujifilm in Japan. Next, please. This is the small intestine liver MPS, one of the results using the device and cells developed by the project. This MPS consists of the 3 parts. First part is the cup insert shown left part in the photo, where FI-seq is plated. This part is responsible for the absorption at the small intestine. After passing through FI-seq, the substance or its metabolism run into the liver part, right side of the device in the photo. Human hepatocytes are plated here, and the chemicals from small intestine compartment are subject to the hepatic metabolism. As a result, we can evaluate the absorption at small intestine and metabolism at the liver, which is so-called first-pass effect, is measured in one device. I don't go into detail of the results shown in right panel of the slide, but this MPS reflects the changes of chemicals well. Next, please. After 5 years effort, 4 MPS has been developed. Then, new project AMED-MPS-2 has started last year. Next slide, please. Again, this is the chart which shows how new test method reaches the regulatory acceptance. AMED-MPS-1 was responsible for the research and development of new test method, in our case, MPS. What is necessary for the next step is to make MPS have good reproducibility and easy handling. These are important for a variety of users to use MPS in their own needs, that is to say, industrial implementation of MPS. To accomplish the industrial implementation of MPS, AMED-MPS-2 project has started last year, following the AMED-MPS-1 project. Next slide, please. This is the process we are thinking necessary for the industrial implementation of the MPS. First part of this process, shown in blue color, is established of the context of MPS use case by user. In other words, performance standards of MPS are considered based on the unmet needs that users are expecting MPS to solve. Next slide, please. In the case of MPS2 project, two model applications are selected. One is first-pass enterohepatic circulation model using MS plate, left one, and the other is small intestinal permeability model employing fluid 3D cross, right one. MS plate, left one, were originally developed by Dr. Matsunaga at Nagoya City University and fit for the evaluation of the first pass effect of the absorption in small intestine and the metabolism in the liver. Fluid 3D cross, right one, were originally developed by Dr. Kimura at Tokai University and distributed by Tokyo Oka Kogyo now. It can apply shear stress by perfusion of culture medium in the upper and lower sides of the membrane. Program 1, led by Dr. Ito at the University of Tsukuba, has selected these two use cases as the model for the implementation of MPS to the industry based on the hearing of farm company about their unmet needs. Program 3, which I organized, is responsible for the standardization of these MPS applications. Let me explain what we are doing in the next slide. Next slide, please. Second part of the industrial implementation of MPS, shown in red color, is extraction and verification of consideration points necessary for the regulatory acceptance of MPS. Selection of the model cells and development of the MPS devices should be according to the performance standards of MPS. To do so, we have to establish the cell criteria and device standards. Cell criteria and device standards are criteria and device standards are consisted of consideration points for each. The example of consideration points for cell criteria are good cell adhesion to MPS, differentiation and maturation of MPS, good functionality fit for context of use on the MPS, variation in cell function, and so on. The example of the consideration points for device standards are evaluation of absorption of compounds, evaluation of effects of plasticizer, cell observability, long-term sterility, sufficient sampling volume, simultaneous execution of multiple experiments, and so on. After the establishment of the test procedures, there are also some consideration points like reproducibility and so on. Here in the following two slides, I will introduce two consideration points, one for cell and the other for device. Next slide, please. This is the result of our paper we published recently. In this paper, we studied the consideration point of commercially available hepatocyte as the cell source for liver MPS by comparing liver characteristics. Left panel showed the distribution of drug metabolism activity in 8 different lots of cryopreserved hepatocyte. As you know, there are several order differences among these 8 lots of hepatocyte. In the right panel, we studied the expression of intestinal markers in human iPS cell-derived hepatocytes. The expression of intestinal markers were observed in some cases, suggesting iPS-derived hepatocytes might have some intestinal characteristics. Next slide, please. Example of consideration points for MPS devices is shown in this slide. Upper left photo is the classical Barak type MPS. As shown in the upper right panel, the liquid going into the chamber makes the laminar flow. Thus, the dissolved oxygen in the medium is consumed by the cell's inlet side, and low oxygen medium go down to the outlet side. As a result, the cells grow in the outlet side are exposed to low oxygen medium. This causes more cell death in the outlet side, comparing to those in the inlet side. This result shows that the medium flow is important to get the good cell culture condition. We are summarizing such kind of consideration point as the application note for MPS users. Next slide, please. Next, let's move to the regulatory side of the MPS development. Next, please. One of our goals is the regulatory acceptance of MPS. That means that the data obtained from MPS is applicable to application of the new drug to authorities. It's not the case in Japan yet. Thus, to get the regulatory acceptance of MPS RS project has started last year, in parallel to the AMED MPS 2 project. MPS RS stands for Regulatory Science, is supported by the Ministry of Health, Labor, and Welfare. Next, please. To fulfill the process of regulatory acceptance requires the participation of 4 stakeholders: academia, supply companies, users, mainly pharma or CROs, and the regulatory sector. In our experience in AMED MPS 1, we found that there are gaps in the understanding and perception of development and use of MPS among these stakeholders at the beginning of the project. Next, please. Thus, the project led by National Institute of Health Sciences was planned, and MPS RS project was initiated last year. As shown in lower side of the slide, the National Institute of Health Sciences, NIHS, conducts testing, research, and studies toward the proper evaluation of the quality, safety, efficacy of pharmaceutical products, foods, and numerous chemicals in the living environment. NIHS is a leading national institute in the field of regulatory science in Japan. Next, please. As I shown the previous slide, the participation of 4 stakeholders, academia, supplier, user, and the regulatory sector is important for the discussion of the way to regulatory acceptance of MPS. Now, we set up the MPS consortium in Japan. The objective of this consortium is promotion of the development of guidelines for test methods using MPS originating in Japan. The consortium consists of 2 parts. First part is for discussion of guideline preparation, including protocol development committee and internalization of committee. Second part is for the discussion among stakeholders. The consortium will hold meeting among the members annually under the planning of the steering committee. The secretariat is located in NIHS. It's just launched, and we are hoping to start discussion not only in Japan, but also with other countries to fulfill this objective, promotion of the development of guidelines for test methods using MPS originating in Japan. Thank you very much for your attention.
Excellent. Thank you so much, Professor Ishida. Very exciting to see all of the activities and efforts going on in Japan, both in the research and the regulatory acceptance space. I have one clarifying question. We have a couple minutes in case anybody else has a clarifying question. Are there any existing examples of new drug applications in Japan that have been supported by MPS data?
Actually, no. The answer is no, not yet. Yeah,
Not yet.
unfortunately. Yeah.
But it's good to have goals, something that you're working towards, right?
Yeah.
That's great. And so you are engaged when you are talking about the users, when you're looking at criteria for acceptance, do the users include both regulatory decision makers and users from, for example, the pharmaceutical industry and drug development space? Do you consider both groups of users?
So there are more users included from the pharma side. And unfortunately, the regulatory side is not so positive to joining for the discussion. But the NIHS, our institute, is just in between the users from pharma and also users from regulatory side. So we are responsible for connecting of these two sectors. Yeah. And we are trying to connect them. Yes.
Great. Okay, thank you very much. So just as a reminder to everyone, please feel free to put questions in the chat with an indication of what speaker the question is directed towards or whether it's a question for the panel discussion at the end. Okay, so again, thank you very much for the wonderful presentation. And now moving on in the agenda, our next presentation is from Jeremy Boydston from the National Biodefense Analysis and Countermeasures Center, or NBAC. He's going to be talking to us about organs on a chip systems development at the NBAC. So Jeremy, I believe the intention is for you to share your own screen.
Yep, you should be looking at it. How are we looking?
Looks fantastic.
Okay. Thank you very much for having me here to speak today. I just will preface that I am at the MBAC, which is a DHS lab. We are not antisocial. I simply do not have a camera, but I'm happy to see all of you who are able to share your lovely faces, but we are not able to. So that's simply it. So anyhow, we're going to talk in two phases today. We're going to talk about where we are developing organ on a chip systems, and then we're going to run through a little bit of work we did with SARS-2 and discuss a little bit how I think we can apply our organ on a chip systems to the studies that we did with animal models. Okay. Disclaimer, we are a highly regulated facility, of course, and all appropriate
Jeremy,
protocols.
I think we have lost your slides.
Oh, no.
You were sharing, but I no longer see them. John, I'll just confirm that that's true.
That's true for me, Nicole.
Okay. Maybe Jeremy, if you could just reshare, please.
All right. Okay, my apologies there. Okay, you have me now?
We see your screen, but not the slides. So we see the Zoom window.
Okay.
Getting
How about
closer.
now?
Yes, maybe just put it into slideshow and we should be good.
Okay.
Switch your displays. There you go.
My apologies.
Perfect.
They had great patience. I got my slides yesterday afternoon, so thank you for your patience there. Okay, so the MBAC is, we have two centers. We have the National Bioforensic Analysis Center. We work very closely with FBI and law enforcement, and we support biocrime investigations. Those are pretty easy to understand. The more scientific mission is that we have a threat characterization center that I work in. We have a group of scientists working throughout all different levels of biocontainment, BSL 2, 3, and 4. And our mission is to provide novel data, empirical data to inform the consequence assessment for modeling bioterrorism risk assessments. And at MBAC, we look at all phases of threats posed by biological aerosols. In particular, I'm an aerosol scientist, so when I look at this problem, I generally look at it from that angle. But we think of the various particle sources, what happens to them in the environment as those particles dry down and decay in the environment and are transported and exposed to a variety of stresses. And then we think also about what happens during the inhalation or the exposure piece. So what factors of that agent or what host factors might increase the probability that any specific host, once exposed, will become infected. And organ on a chip is a technology that we believe will suit this, be one tool that we can use to help answer this question. So we know from experience that animal models might not effectively translate to human systems. Oftentimes when we do studies in vitro, it's a singular cell type. And when we have an organ on a chip system, this audience knows as well as or much better than my average audience on this topic, that you need that complexity to accurately mimic. So you need that physical stress induced by movement and breathing to truly mimic a animal system. We are currently using the emulate system, and we're in the process of acquiring the tissue system. So what can we assess via an aerosol model or an inhalational model? So we can look at different cell types, different particle sizes, agents, strains, what is carrying them from an aerosol scientist point of view. It is a very diverse number of medias and proteins. Could it be mucus containing from the upper respiratory tract? Or is it lung surfactant from the lower respiratory tract from truly small particles? How do those influence infectivity? And again, how do host factors and the host response to infection mediate that probability that any singular host becomes infected or a population? So one thing that we've been looking at recently, we've been doing some work in looking at host factors that influence the probability of infection. So what in the environment might cause a host to be more likely to become infected? And so here I'm highlighting mucociliary clearance in really the upper respiratory tract, not the lower respiratory tract. When you think of seasonality in viral infections, we've known for quite a long time that a number of different viruses, influenza, RSV, and coronaviruses have been demonstrated to have some seasonality. And one of the theories is that humidity, really it's absolute humidity or changes in absolute humidity. But that change in humidity is what drives that seasonality. It's not that you're outside or inside more. It's really your health, your respiratory tract health as the seasons change. And so when we have dry air, such as we do in temperate climates, we have a bit of a inhibition or an impaired mucociliary clearance or mucociliary clearance deficiency in the upper respiratory tract caused by the inhalation of low humidity air. That can also cause, the low humidity air can also cause some innate immunity defects or impaired barrier functions. And so we tried to ask the question, can we see this phenomenon both in vivo? So is this something that would be agnostic to infection and we'd be able to see differences regardless of the presence or absence of a pathogen? And how does that affect the dose response relationship? So really quickly, we use this mucociliary clearance assay where we euthanize an animal, collect tissues from the upper respiratory tract, specifically the trachea. Within 5 minutes, we take that tissue and we mount it onto a slide with some 200 nanometer or 0.2 micron fluorescent beads. And then we're able to measure the background. I didn't want too many slides here, but what we do is we precondition these animals at either normal humidity or in a dry, very arid environment, 10% RH to be specific, for 1 week prior to doing this assay, or we have normal controls. So we're saying, does that mucociliary clearance, the ability of particles that deposit in your upper respiratory tract, are they impacted, that clearance rate, by dry air? And just briefly, what you're seeing here is just how we process this data. So on the left, you're looking at the raw data, and that twitching you can see is actually the ciliated cells beating along that surface, pushing those deposited particles up and moving them out of the respiratory tract where they'd become ingested. We then go through a spot detection and then do a frame-to-frame linking and particle tracking, which you can see on the right. And this allows us to measure very precisely the rate of particle clearance, and we can look at how different conditions affect how quickly these particles are cleared. So here you can see the influence of humidity on this clearance rate. And what you'll see, if you look on the right, you see some particles are not tracked, the majority of them are, and I'm not going to do a dive into the statistics here, but what I can tell you is we can prove with very solid data and statistical measures that if you look on the left in the Brownian motion, that's just background. These particles are just kind of waddling around, they're not moving much. Animals that have been in dry air for 7 days, they have a slower rate at which they're able to cause or move or clear these particles. And animals that are at normal conditions, and we can see this in both mice and hamsters, are able to clear the particles from the respiratory tract most rapidly. And so what does this mean? Again, we believe, and the data that are in the literature demonstrate that these host factors are important things that we should be looking at to determine what is the likelihood or the probability that any host might become infected. And Emulate has actually shown this. Now, they haven't quantified it and shown humidity effects. This is a project that we are working on right now, where we are hoping to use these kind of systems to demonstrate that you can see similar effects. So what you're looking at on the right is where they showed a different fluorescent particle moving. So you can see it moving across the cell surface or the ALI. We haven't gotten there yet with the humidity study, but that's something that we're working on, and I think poses an opportunity where we can look at these host factors and we can change different things about the environment that the organ on a chip cells are in, and then can we measure changes based on that? Of course, there are a lot of challenges for aerosol deposition when you're looking at an organ on a chip system. So animals, mammals, we are deposition machines, right? As we inhale and we breathe in and out, that allows particles to deposit by our natural breathing. Organs on a chip do not have that. And it is a great challenge because of the small passages that are typically involved in these systems. So we're looking at trying this with a few different chips here, some of the chips that you're all familiar with, both the Emulate S1 chip. We've tried out the Emulate OpenTop chip. I think this would work quite well with the Tissues Humimic system. And one of the things we're looking at using here is an electrodynamic balance. This has been developed in collaboration with the University of Bristol, also with Distal, who is a kind of counterpart to the MBAC in the UK, and IWTSD. This is a particle trap, and what it does is it allows us to generate particles, hold them in electrodynamic balance, and then shoot them down. If you were to look at this slide, you can see there'd be a droplet trajectory. They're captured in these ring electrodes, and then you can kind of shoot them down into a test area that's around 1 centimeter in size. So it'd be perfect for organ on a chip studies. Here's a video. If you look, you can see these particles. This is slowed way down. They look bigger than they really are because they're vibrating, but they come into the ring electrodes. They're held there until we reach equilibrium. Of course, this poses an environment where we could look at the stability of these aerosols over time. And then after either an aging process or if we just want to look at them right away, and there you can see that they dried down, we can then shoot them straight down without having to blow a bunch of air onto the cells, which, as you can imagine, is going to kill most of our cells. So the Celeb system, or the Celeb's particle levitator, is something that we're looking and actively working to develop to deposit aerosols onto an organ on a chip. So, that is another, just to say in a broader text, what effects of age aerosols can we find in activity? So that is something that when we trap them, whether it be in a celeb system or in one of the other environmental decay systems that we have, we do see a lot of utility in organ-on-a-chip technology to look at the effects of age aerosols on infectivity. And what are those factors that influence the decay of biological particles in the air that we might be able to see with an organ-on-a-chip and we might not see in a traditional plating method or cell-based method. Okay, we went through all this. In the interest of time, I'll just reiterate we're actively seeking to develop aerosol deposition systems for organ-on-a-chips. However, even though you're getting the angle from me, the aerosol guy, we're also looking at other systems for the toxins. There's a whole lot of other things that would be interesting, a variety of other tissues we're hoping to develop. And so I'm going to run through some slides that demonstrate what I think we'd be able to use these organ-on-a-chip systems for at the NBAC once we get them up and running in a little larger throughput. So recently we looked at the dose response relationship for SARS-2, and we looked at a couple different isolates or variants. We looked at Washington 1 versus alpha or B117. We use golden Syrian hamsters, and what we looked at is in an aerosol model of COVID-19, is there a difference in infectivity in Washington 1 versus alpha? And so as most of you would know if you've done any SARS work, which is probably everybody here, the hamster is a very good model for COVID-19 inhalational models. If you look at the bottom of the slide there, you can see an alignment, and it's primarily due to just conservation in ACE2. But we've found it to be a very good model for inhalational COVID-19. This is, again, an aerosol system. Here's just a quick look at it. We generate aerosols using a 2-fluid nozzle. The one we use is called a Lee nozzle. It generates small particle aerosols. Those are kind of the, generally, the particle size that we worry about for transmission are generally the smaller particle sizes. And that is used to be representative of what might be generated and persist in the air for some period of time by infected people. The hamsters were singly housed and exposed for 10 minutes. Our group size was 8. We did 6 groups of 8. So particle generation in the respiratory tract, I just want to kind of remind you how, or give you kind of the look of how we look at it. We think of this particle generation, and while you hear of these cough-generated aerosols, and those are generally not present in the air for a very long time. Those are usually in the 30 to 100 micron range, and they don't persist for a very long time. In general, just breathing alone can generate small particle aerosols that are infectious. And here you can see a particle size distribution that demonstrates most of those particles that we're generating when we're infected are generally in the small size range. We measure body weight activity as far as running whale activity and respirations in these animals pretty much every day. It is every day while they are infected. And then we also look at post-exposure sampling. Our final measure for infection is seroconversion via ELISA. We measure temperature, and we also look at swabs for both infectious RNA and a TCID50, which is similar to a PFU assay, and viral RNA via PCR. And what we could see here is that observationally, both Washington 1 and alpha had similar magnitudes of infection in these animals compared to the uninfected control. So we saw a decrease in activity that peaked on day 6, and it was similar for both viruses. The increase in respiratory rate was similar for both viruses. And the rate of weight gain decrease was similar for both alpha and Washington 1. Lastly, the shedding of infectious virus was similar for both alpha and Washington 1. We generally detected right on day 2, kind of peaking on day 4 to 6, and then continues out to maybe day 8. And what we could see when we did a proband analysis is that there was not a statistically significant difference in the median infectious dose between Washington 1 and alpha. So if we applied this model to organ-on-a-chip, we would likely be able to generate data probably a little bit quicker. Now, we don't have the full complement of the mammalian immune system, but is this the kind of study that we might be able to do in organ-on-a-chip technology? It's something we are working towards. And lastly, I have just a few slides on how aerosol particle size influences the amount of agent it is required to cause infection. So it's well known that as particles are smaller, it generally takes less of that agent to cause infection. And so that particle size affects the transport of particles in the environment. So when we look at two different aerosol generators, both that small particle generator and a larger particle generator, we consider 5 microns to be relatively large in a hamster. And you can see the two different particle size distributions. We're asking the question in alpha, do we see a difference in the probability of infection with the small versus the large particle aerosols? And in fact, we do. We see a very large difference. This is approximately 30-fold. So the ID50 for small particle alpha is about 0.3 TC-ID50, or kind of less than 1 PFU. For a larger particle aerosol, it's around 10 TC-ID50, or like 10 PFU. And so what we could find is that with larger particle aerosols, the probability of infection is substantially lower. And so in summary, the infectious dose was significantly different when we looked at the influence of particle size in the alpha model. We did not see a difference between Washington 1 and alpha. And we're looking at how can we combine these organ-on-a-chip technologies where we'd be able to control that complement as the change of cell tropism between the upper respiratory tract and lower respiratory tract. Can we do particle size studies in organ-on-a-chip systems where we'd be able to mimic both the upper respiratory tract and the lower respiratory tract and tune the particle size to fit the model we are looking at? And with that, thank you for your time, and I am happy to take any questions.
Fantastic. Thank you so much, Jeremy. That was really great. Do we have 1 or 2 minutes for any clarifying questions? I have probably a very sort of simplistic question. The initial work that you showed on the influence of humidity, is that it seems like it's consistent with kind of the seasonal variation of infectivity that we see sort of during more humid versus less humid months. Is that accurate?
Yeah, absolutely. And so when I've got a ton of time, I can walk you through data, but there are some data that are published that show that it's inversely related. So the humidity is inversely related to the rate of influenza in humans in the United States, right? So during the summertime, less flu, they've looked at all the different measures, right? Is it sunlight? Is it absolute versus relative humidity? Vitamin D, time outdoors. The fact of the matter is we spend the vast majority of our time indoors. And so yeah, as it gets more dry and more potently, right when a cold front comes in, if you study this, it's been studied in school children, within those few days, it's really kind of like 5 days to 7 days, right after you have that change from nice humid air to very dry air, you actually have a spike in influenza. And it's believed to be, and statistically shown to be, most likely to be related to humidity. So, what we're working on right now in the hamster model, we haven't seen any big differences yet, but it doesn't mean that they're not there. And we're still actively pursuing the humidity and host response piece.
So another data point for me, having humidifiers in my kids' rooms. So, okay.
Absolutely.
One question in the chat from Donningbert: have you tried to expose the open top emulate chips in your hamster aerosol exposure chamber?
No, we haven't. Now, we have a similar system that we're planning to do in the coming year. It won't actually be the hamster one, but it'll be very analogous. And so we're going to look at, that traditional kind of gravitational settling of aerosols. We're going to look like, and we may or may not use the Vitrocell, but some sort of a inertial impaction method, something like that. And then we're also going to try the celebs. But I've spoken with some people who have tried, flowing air over them, and you kind of blast. You can't put too much air over the aerosols. You'll kill the cell so quickly. So we're really hopeful that either the system you're talking about, Don, or the celebs will be suitable to maintain cell viability, but have a accurate and quantifiable amount of aerosol deposition.
Great, thank you. Okay, so keep the questions coming in the chat, everyone, and we'll now move to our next presentation. So Tyler Goralski will be speaking to us on Soldier on a Chip, interrogating the effects of biological threat agent exposure using a multi-organ MPS. So Tyler, we'll just get your slides up and hopefully, if need be, perhaps Tyler can share his own slides.
Yeah, I'm good to go if you need me to share.
Let's try this one more. There we go. Perfect.
Okay, great. All right, well, thank you, Nicole. So I'm Tyler Gorowski. I'm a research biologist with the DevCom Chemical Biological Center Molecular Toxicology branch of the Threat Agent Sciences Division. So for this talk today, I'm going to be giving you a brief overview of my group, kind of what we're focused on, as well as an overview of some of the MPS models that we have in place, and then really focus on our efforts to implement a multi-organ chip system in order to assess the toxicological effects of biological threat agents in this case. Next slide, please. So this is just a disclaimer that the views expressed in this presentation are mine and do not necessarily reflect that of the DoD, the Army, or the US government, and any use of trademark names also does not imply endorsement by the US Army. Next slide. Okay, so just to give you a little bit of background on my group, so the molecular toxicology branch, we're really focused on predictive toxicology. So we use in vitro systems, whether we develop them ourselves in-house or we acquire them or adapt them to our purposes to test the toxicological effects of chemical and biological threat agents that we can then use to guide our downstream in vivo assessments, hopefully using less animals as a result. So here I'm showing you a list of different operational tissue systems. You can see we have a number of different means of performing these assessments, whether that's 2D, 3D, real-time cell analysis, organoids, and most recently we've been adopting these different commercial MPS models. You can see we're still in development with these brain-on-a-chip or blood-brain barrier chip, but we do have a number of different organ systems up and running using the various commercial systems. Next slide. Okay, so Don Ingber published this really fantastic image here that really showcases the breadth of the different types of MPS models that are currently being used. If you want to hit the next one time. So here I'm just pointing out the three general types of MPS models that we actually use in-house, and that would be the emulate system, the CM Bio system, and the tissue system. So over the next few slides, I'm going to go through those systems and kind of give you an overview and on the tissue system, and then go into those data that we've generated for our current study. Next slide, please. One more. Right, so I'll present these kind of, the way we like to think about it is increasing levels of complexity. First, I'm going to highlight the CN Bio system. Next slide. One more time. Okay, so this is showing you on this slide these CN Bio physiomimics. So this is a plate-based MPS model, which I'm sure you're going to hear more about by the from the CN Bio talk later. So there's 12 wells per plate. Within each well is a transwell containing a scaffold. So within this scaffold, in our hands, we actually seed hepatocytes. We're using this model for liver. One more time on the slide. And so when you see these hepatocytes into the scaffold, they form these 3D structures to have that 3D architecture. The plates themselves have circulating microfluidic flow. So we have that shear stress on the microenvironment, and we have a more physiologically relevant system here than just looking at two-dimensional hepatocytes on plastic. We can keep these cells going out to a month, and we use these really as a first pass for toxicity. So any emerging threats that come down the line that we're interested in liver toxicity, we would pass it through the CM Bio physiomimic system first. Next slide, please. One more time. Okay, and now I'm going to showcase the emulate system. Next slide. So here's an overview of the emulate chip. I'm sure a number of you here are pretty well versed in this system, but I'll go over it briefly. So if you hit next like 4 times. Yeah, so here I'm just highlighting the different components of this chip. It's a 2-channel system. The top channel, which is where you would seed your epithelium, is separated from the bottom channel by a permeable membrane. Your bottom channel would be where you would seed your endothelium. There are vacuum channels on either side of the chip. So in the case of the lung or the gut, you can recreate simulated breathing or undulation. And within this system, you really can increase the level of complexity if you like. You can do co-cultures or up to even quad cultures in the case of the liver. So with the liver system in our hands, we actually see the top channel with hepatocytes, and then in the bottom channel, we see liver sinusoidal endothelial cells, stellate cells, as well as Kupffer cells. So currently, we have this system operational for the liver, the alveolus, the small airway, and the renal proximal tubule kidney, and we're working on getting the brain system in-house now. Next slide, please. One more time. Right, and then finally, I'm going to discuss the tissue system. So this is the most complex system that I think we have in-house, and I'll be showing you data using this system as well. So next slide. All right, so here's an overview of the Humimic multi-organ chip system. Here in the top left, I'm showing you the chip 3. So there's 3 different compartments. This is just a polycarbonate chip. Underneath the compartments is a microfluidic layer that's formed by PDMS, and then finally a microscope slide underneath so you can view the cells in real time. These chips are all run by microfluidic pumps that introduce pulsatile flow. So it'd be an exchange of vacuum and pressure that pushes the media through the channels into the compartments of the whatever organ type you're interested in. Next slide. We're also using the chip 4 system. So this is a bit more complex. There's 4 organs that you can add to this system at one time. Next slide, or next. One more time, thanks. So the really neat capability of this system is that it's actually separated into 2 different circuits. So here on the left, I'm showing you an overview of the chip 4, and there is an excretion circuit or the kidney circuit, as well as a blood circuit. So a barrier is actually formed by seeding renal proximal tubule cells. You can see there's a simulated loop of Henle, as well as glomerular cells. So that forms that barrier that keeps the blood circuit from the urine circuit separate. And this system is really nice in that you can do ADME type studies. And the larger number of organs that you're using within this chip allows you to look at more subsequent effects to different tissue types. And just to highlight that, down at the bottom of this slide, this is actually showing cardiac organoids from the chip 3 system, where the chip was dosed with mustard HD in the lung compartment, and was allowed to run under flow for 24 hours. We then looked at the cardiac organoids, which never actually received the HD directly. And if you go next slide, or next. And we started to see cell death within the cardiac organoids. So this is a live dead stain. The red cells are dead, the green are alive. And you can see that the organoids are starting to come apart. So this is one of the really nice utilities of this system. So you can dose on one organ type and then look at the effects downstream and see how that's being affected, even though that wasn't the site of the exposure. Next slide, please. So here's kind of an overview of the study that's currently ongoing using the chip 3 system. So within the chip 3, we have seeded cardiac organoids, lung tissue, and skin tissue. We're exposing that lung to various chemical and biological threat agents, allowing that exposure to progress through the chip so that the other organs would be seemingly affected by that agent. We're then extracting the tissues and then performing downstream multi-omic analysis. So in this case, we're doing metabolomics and proteomics. But just to highlight, this system, as well as the CMBio and Emulate2, are amenable to a number of different downstream analyses. They're really great for staining in this case. The effluent can be collected in real time as you're doing your studies and do a number of different biochemical downstream assays, electrophysiology on the cardiac tissues, and more. So next slide, please. So for this talk, as I mentioned, we exposed to a handful of different chemical and biological threat agents. but I wanted to focus on the Staphylococcus enterotoxin B exposure. This is just showing you a breakdown of our proteomics data. So in the table in the top is showing the number of high confident proteins that were identified in each of the 3 different types of tissues, as well as the significantly changed proteins. We were able to generate volcano plots for each of the 3 tissue types and show the statistically upregulated and downregulated proteins as compared to the untreated chips. And then, of course, we can generate heat maps for all of the proteins that were identified within this experiment. Next slide, please. So for the sake of time, I'm going to highlight one of the more interesting findings that came of this proteomics data. And bottom line up front, this allowed us to identify potential biomarkers for exposure to Staphylococcus enterotoxin B that were previously unknown with this particular agent. So in the top graph here, I'm showing you the relative abundance of this sumo-conjugating enzyme UBC9. And so for those that aren't aware, might be unsure about SUMOlations, this stands for small ubiquitin-like modifying protein. And this protein, like ubiquitin, is involved in the regulation of a number of different cellular processes. It's usually repression of a number of these processes, which are shown down here in the bottom. But this sumo-conjugating enzyme UBC9 is absolutely required for SUMOlation to occur. And here we're showing that it's downregulated compared to the PBS controls. So we went through the data, looked at the enrichment mapping, so all the different biological processes that were highly dysregulated compared to the control chips, and searched for a number of these processes that are implicated in SUMOlation that are shown below. If you go to the next slide. So we did find that this FIS1 protein was also dysregulated. So this was up compared to the BBS control. And this FIS1 protein is required for peripheral division of mitochondria. So in the last slide at the top right on that figure, mitochondrial fission and fusion is implicated in SUMOlation. And so these processes occur under cellular stress. This can be due to the environment, or potentially in this case due to Staphylococcus enterotoxin B exposure. But when mitochondrial fission and fusion can't occur, the cells become sick, and because the mitochondria are affected, you're really hurting the downstream energetics as a result of the lack of fusion and fission. So we had identified this protein and decided to kind of go down a rabbit hole here and see what other mitochondrial dysfunction we could identify. Next slide, please. So when looking at the enrichment mapping, so again, this is looking at dysregulated biological processes as compared to the control chips, and just searching for mitochondria, you can see with all 3 different tissue types, there was a number of different mitochondrial dysregulations. So really, at this point, this is worth looking into further. We want to do more experiments to see exactly how the mitochondria are being affected after exposure. We have a mitochondria expert in our lab that has a few different techniques that we could implement to dig further into this. But if you go to the next slide, I'm breaking it down a little bit further by the different tissue types. So now we're looking at enrichment mapping for each of the tissue types that were the most dysregulated. And if you look at the most up and down regulated proteins in the skin, the ones that I've highlighted are directly implicated in SUMOlation. So SUMOlation target proteins are upregulated, which you would expect. The one above that just says SUMOlation, when you dig further, that is also target proteins. And then downregulated are a number of different signaling pathways that you would expect to be up if SUMOlation was functioning appropriately, but they're all down compared to the control. Next slide. Next slide, please. And then again, if you look at cardiac, we're seeing a similar thing here. more signaling that was way up that should have been down. We see T cell activation, which is actually the mechanism of action for SEB. And then the fast signaling pathways. So fast associated apoptosis is actually required for mitochondrial fission. And with that being way down, we would expect that that would be having an effect. And finally, next slide. When you look at the lung, we again see markers that would be implicated in simulation, but interestingly enough, we also saw the upregulation of another number of markers or proteins that would be implicated in potential biomarkers. So with glycolysis and pentose phosphate pathway being up, this is indicative of a switch over to anaerobic respiration. So we can look at things like lactic acid, ATP synthesis as biomarkers. Vasopressin synthesis is also up, which is directly correlated to blood pressure. And then obviously with adrenaline and noradrenaline biosynthesis being up, that's something else we could look into. Next slide, please. So for the next couple of slides, I just wanted to highlight some of the other applications for this multi-organ chip system from Tissues that we're going to be using in-house. First, I wanted to present this inhale system. So this stands for Independent Holistic Air Liquid Exposure System. This is developed by Philip Morris International. And what they were able to do is create this inhalation system that recapitulates the entire airway. So there's an oral model, a lung model, and an alveolus model. And you can look at the maturation of the aerosol as it goes through the lung. It's temperature controlled based on where you are in the lung, as well as humidity controlled. Next slide, please. So PMI, they've been working with Tissues to integrate the Tissues chip into the inhale system. So they were able to take this chip and integrate that at various portions throughout the inhale system so that you can do real-time sampling of the tissues at those different regions in the airway. So we're getting this system in-house with the Tissues chips, and we're going to be beta testing this here in late summer. Next slide, please. And then finally, just some additional multi-organ chip projects that we have ongoing in-house. The first would be an ADME chip, MOC. So with this chip 4 system, as I kind of mentioned earlier, you can dose this with agent or compound, look at absorption, distribution, metabolism, excretion. Tissues is working on a program to do all the analysis for that. And in this case, what we'll be doing is trying to establish a bacterial infection model with this chip and then dose that with different antibiotics that are known to be efficacious towards those infections and see how that compares to some of the clinical and in vivo data that's already out there. So we're working on doing this already with MRSA, and we'll be taking this in the BSL-3 to do YP. We'll also be working with PNNL to look at VOC coming off of the lung compartment of these multi-organ chips. So they've gotten into the space of breath mass spec, and we're interested in seeing, can you detect VOCs from these chips, and how does that compare to human breath, what we are able to detect in the chip. And then finally, as we've kind of been putting the cart before the horse, we're finally getting to the point where we're going to dose the chips with known drugs that are well characterized with omics, and then see how those omics data generated in the chip compared to that which is in a human. Next slide. Okay, and this is just to sum up all the different systems that we have in-house. I'm just highlighting here the different commercial off-the-shelf systems that we are using just for the predictive toxicology space. And the next slide, one more time. I just want to thank my team here. So all these people listed here to the left have played an integral role really in all of the different MPS efforts that we've had ongoing. And with that, I think I'm close to time, so I'll take any questions.
Perfect. Thank you, Tyler. Right on time. Any clarifying questions for Tyler? I had one question about the biomarkers that you're identifying. Is there any potential or intention to potentially operationalize some of those in a way that could be sort of field deployable for like triage situations or treatment identification?
Yeah, so that's the intention for the funding agency. They're really hoping to identify something that's going to be translatable to some type of wearable sensor. They're really interested in early detection of either the chem or the biological exposures, and then see if they can translate some of these biomarkers to physiological effects.
Great. Another question, how is the performance of the inhaled system for deposition of pathogen in the tracheal region versus alveoli, et cetera?
Yeah, so we have definitely not gotten that far yet. That's something that's going to be tested way down the road. We're starting out with chem first, and then we'll be switching to bio. There's, on our end, the way we have to operate, there's also some safety concerns with the types of agents that we work with. So all these things have to be figured out before we can even get that far to do those agent exposures.
Okay, great. Thank you. So keep the questions coming in the chat, and for the sake of time, we're going to move to our next presenter, who is Dayton Pettibone from the US FDA, and he will be speaking to us on evaluation of testicular organoids as a model for Zika virus infection. So Dayton, we can see you and hopefully hear you.
Okay, great. Thank you. Thank you for the introduction. So the outbreak of Zika virus disease in the Americas demonstrated how a relatively obscure and mild mosquito-borne disease can become a global health emergency, with infections resulting in congenital Zika syndrome in unborn children. Interestingly, in addition to being a mosquito-borne virus, Zika virus can also infect the male reproductive tract and be spread sexually outside of endemic areas. To address Zika virus sexual transmission, my lab is developing and evaluating testicular organoid models using primary cells from the rhesus macaque that can be applied to non-clinical in vitro testing of anti-Zika virus therapies. Ultimately, this system might support the 3 R's through its use in preventing the spread of Zika virus and other infectious diseases. Next slide, please. So the Zika virus is a blood-borne flavivirus first identified in Uganda in 1947 in the rhesus macaque monkey. It was later identified in humans in 1952 in Uganda and the United Republic of Tanzania. Since then, outbreaks of Zika virus have been reported in tropical Africa, Southeast Asia, the Pacific Islands, and the Americas. Zika virus is spread predominantly through the human population by mosquitoes. Interestingly, in addition to being a mosquito-borne virus, Zika can also infect the male reproductive tract and be spread sexually. There is no evidence of sexual transmission for any other arthropod-borne flavivirus, making Zika virus unique in this respect. So Zika virus can also remain in the female reproductive tract for several months and be vertically transmitted to the offspring, resulting in negative pregnancy outcomes. So next, please. I'm sorry, go back. Okay, so our development of MPS to evaluate Zika virus infection and anti-Zika therapies will focus on its sexual transmission, specifically by using rhesus macaque primary cells from the testes to generate testicular organoids as in vitro models of Zika virus infection. Next, please. In most people who become infected with Zika virus, the symptoms are generally mild, but there is an increased risk for Guillain-Barré syndrome, which is a neurological condition in which the immune system attacks the peripheral nerves and can result in muscle weakness and sometimes paralysis. The 2015 to 2017 outbreak of Zika virus revealed infection could cause severe clinical manifestations for fetuses and newborns. Collectively, these are referred to as congenital Zika syndrome, which is a distinct pattern of birth defects and disabilities, including microcephaly and pregnancy complications such as fetal loss, stillbirth, and preterm birth. Microcephaly is a condition in which the infant's head is significantly smaller than expected, often due to abnormal brain development. Symptoms vary and include intellectual disabilities and speech delay to seizures and abnormal muscle functionality in severe cases. At present, there is no FDA-approved vaccine or antiviral medication, but numerous therapies are in development. Zika virus is also listed by the WHO as a priority disease that poses the greatest public health risk due to its epidemic potential and insufficient countermeasures for an estimated 5 to 15% of infants born to Zika virus infected mothers that have evidence of Zika-related complications. Next, please. So why are we developing non-human primate testicular organoids, which I abbreviate here as NHPTO? Well, the wild-type rodent cells are generally refractory to Zika virus infection. This is because the Zika NS5 protein does not inhibit the STAT-mediated interferon antiviral signaling to establish infection within the cells. Therefore, rodents must generally be infected following gene knockout, such as the interferon alpha beta receptor, or following drug-induced immunosuppression. And while significant mechanistic data pertaining to Zika virus infection and pathogenic effects have been collected using modified rodent models for Zika virus infection, the responses in these rodents is generally much more severe than what is observed in humans and in non-human primates. However, the Zika virus infections in the rhesus macaque also will infect the male reproductive tract and exhibit similar pathologies to humans, including the pregnancy complications. Also, reservoirs of Zika virus infection in the testes might complicate development of therapeutics, and proposed anti-Zika antiviral therapies should be evaluated for their ability to clear persistent infection in the testicular testicular cells once this model is more mature. So using the non-human primate testicular organoids to evaluate the anti-Zika countermeasures might also reduce the demand for in vivo non-clinical safety and efficacy studies in non-human primates. Next slide, please. So to generate the testicular organoids, testes from a single macaque were collected on a separate completed study, and the cells were enzymatically dissociated from the testis tissue. These cells were then exposed to Zika virus or mock infection control. These cells were then seeded into a microwell system. Advance once, please. Shown here in a 24-well plate format, with each cell having 1,200 microwells to generate the non-human primate testicular organoids. Formation of the testicular organoids reveals that the testicular cells have coalesced to form compact 500-cell organoids following 2 days of culture, and that they can maintain their morphology, averaging 100 microns in diameter, as shown here on days 5, 7, and 9. One more click, please. So the Zika virus infected and mock-infected control organoids were then cultured for 21 days and sampled at predetermined time points. The Zika virus RNA was analyzed in both organoids and the supernatant, and the supernatants were also used to measure changes in cytotoxicity, testosterone, and inhibin B release, as well as cytokine levels. One click forward, please. So briefly, before I discuss the Zika virus infectivity findings, I'd like to mention that we evaluated the testicular organoids, and we identified that they contain gene expression markers for Sertoli cells, Leydig cells, spermatogonial stem cells, and undifferentiated germ cells. We also assessed the viability as measured through ATP levels, which revealed consistent ATP levels over the 21-day culture interval. And the testicular organoids were also able to respond to cytotoxic stress over the 21-day culture. Next slide, please. So we demonstrated Zika virus infectivity in the non-human primate testicular organoids after optimization of the infectivity assay. The optimization was performed using a broad range of infection conditions, covering an MOI, multiplicity of infection of 0.25, which is one infectious virus per each 4 cells, up to an MOI of 14, which is 14 infectious particles per cell. And then we evaluated the results for consistency and detection of the viral RNA. The MOIs of 3, 5, and 7 were subsequently performed because the results were consistent with acceptable fluctuations. Infectivity evaluated through 21 days of culture revealed that the Zika virus RNA detection was reproducible and sustained throughout the culture interval in both the supernatants and in the organoids at levels of 1 × 10⁻² to near 1 × 10⁵ copies per mil throughout the culture interval. And at several times, the Zika virus half-life, which is with its low thermal stability, is about 11.8 hours in culture at 37 degrees, demonstrating that the Zika virus is replicating within the testicular organoids. Next slide, please. So the deleterious effects of Zika virus in the testicular cells was evaluated by measuring the cytotoxicity and testosterone and inhibin B as indicators of metabolic function in the Leydig cells and Sertoli cells, respectively, over the 21-day culture. Cytotoxicity was measured as release of a cytoplasmic enzyme from damaged cells into the culture supernatant, and as shown here is the ratio for infected testicular organoids for all MOIs as compared to the mock infection controls. We found the cytotoxicity levels remain consistent, which is expected as Zika virus has demonstrated the ability to infect human and non-human primate testicular models and in vitro Sertoli cells without a cytopathic effect. Testosterone production also remained consistent throughout the infection in all MOIs and was not significantly different from the mock infection controls. However, there was a significant effect of culture time on testosterone accumulation between days 2 and days 5 and 9 through 21 of culture, indicating the Leydig cells in the testicular organoids are metabolically active. Likewise, inhibin B release remained stable across the MOIs, but the inhibin B level significantly decreased with time as compared to day 2 of culture, indicating possible decrease in Sertoli cell metabolic activity. This might be due to a lack of follicle-stimulating hormone in the media to stimulate inhibin B production. These findings agree with previous studies that indicate testosterone and inhibin B levels were minimally affected in Zika virus infected human testicular explants, while the in vivo non-human primate and human testosterone serum levels following Zika virus infection are somewhat mixed, with some studies indicating no change in levels and other studies suggesting decreased testosterone and inhibin B levels. Next slide, please. So to further characterize the Zika virus infection in the non-human primate testicular organoids, cytokine secretion levels in the supernatants were evaluated using a panel of 27 cytokines. Here, a 2-fold or greater change in secreted levels or p-value less than 0.05 was considered significant. Overall, Zika virus infections altered cytokine secretions, both increased and decreased these levels as a function of time, which peaked at day 9, shown in the lower left corner, before the number of altered cytokines began to again decrease. We observed an imbalanced inflammatory cytokine response and increased pro-inflammatory cytokines such as IL-6, IL-8, and IL-15 without increased secretion of anti-inflammatory cytokines such as IL-1, IL-4, or IL-10. Several adaptive immunity cytokines also had altered secretion following infection, such as VEGF and RANTS. We also observed a biphasic response to several of these cytokines, including IL-6 and RANTS, where there was a decrease in secretion levels, as seen for IL-6 on day 5, the upper center graph, and RANTS on day 9, the lower left graph. followed by increased secretion observed for IL-6 on day 7 through 21, and for RANTs on day 14, which is shown in the lower center graph. Suppression of these cytokines has also been observed in human Sertoli cells and several neural cell lines exposed to Zika virus. This pattern might partially be explained by Zika virus suppressing cytokine signaling in the testicular organoids during an acute infection versus recovery phase of infection. Additionally, some of these cytokines, such as RANTs, which has been shown necessary for persistence of Zika virus infection in several human cell lines, might provide anti-Zika virus therapeutic targets for clearing infection. Next slide, please. So to summarize the development of the testicular organoid models of Zika virus infection, the testicular organoids contain key cell types found in the in vivo testes. The testicular organoids were also suitable for detection of ATP, cytotoxic responses, testosterone and inhibin B release, and cytokine secretion. Zika virus replicated and was detected within the testicular organoids and in the supernatants. Zika virus infection in the testicular organoids did not alter cytotoxicity or affect testosterone or inhibin B release, and modest alterations in cytokine secretion were observed. In conclusion, these data demonstrate the testicular organoid's potential for non-clinical studies of Zika virus infection. So this work is really still in its early stages of development and evaluation. And during these studies, we are trying to see what types of information we can obtain from these models. So next slide. So potential applications for the testicular organoid models to support medical countermeasure development would be that the testicular organoids might find use in non-clinical settings by expanding therapy testing for rare diseases and to establish strategies to reduce demand for non-human primates. The key benefit of testicular organoid development that they provide to regulatory science is laboratory models to expand therapy approvals for infectious diseases, to assess pathogenic infections, and to complement animal studies through development of study designs, candidate therapy and dose selection, or interpreting toxicity data for organ-specific toxicity before committing to studies in animals. The models may also provide cross-comparisons of in vivo animal and human data, which could identify species-specific effects and build confidence in human in vitro models for regulatory use. So next slide. Okay, so moving forward, we will establish a very simplified model of Zika virus sexual transmission using the tissue's organ on a chip, shown here with 2 organ compartments linked by a microcircuit circulating a common media between them. The chip will contain Zika virus infected testicular organoids or mock infection controls, as well as non-human primates neurospheres as a target of Zika virus infection. The microcircuit will circulate Zika virus in a common media between the compartments, and we can monitor Zika virus production, infectivity, cytotoxicity, metabolic function, and their ability to respond to toxic agents. We will further evaluate the anti-Zika countermeasures in this microfluidic system. Next slide, please. So briefly, because I work with non-human primates and to further support their use in research, recently the National Academy has released a report on the status of NHP models in biomedical research and found that the COVID-19 pandemic, along with the restrictions on their exportation and transportation, have greatly limited the ability of non-human primates that impact public health and national security. The report also suggests that NAMs such as organoids, organ on a chip, and in silico methods could be used for improved could be used to improve the translatability of non-clinical research, to extend current knowledge of human diseases, and to address shortages or replace non-human primates in research. And while the study I am presenting here today is still in its nascency, it is an example of supporting the principles of 3R in practice, and developing the non-human primate testicular organoids potentially provides a possible bridge along with similar in vitro models for improving translation of non-clinical data into human clinically relevant data. So next slide. And I'd like to thank my colleagues at FDA, both at NCTR and at CBER. The analysis of Zika virus was done in the lab of Maria Rios at CBER, and I'd be happy to take any questions you have.
Great. Thank you so much, Dayton. Really fascinating talk. Couple of questions in the chat that have a similar theme. So first, great talk, Dayton, and this is from Simon Funnell from the UK Health Safety Agency. Are you considering looking at human testicular organoids? And if not, is this because of lack of access to donor tissue?
That's precisely why we're not looking at them. We were trying to acquire some of those materials, but they are difficult to get your hands on. So the non-human primate provides a model that's relevant for studying Zika virus infection and is more readily available to procure than are the human testicular tissues.
Okay, thanks. And that also addresses the question from Emily Richardson about the sort of extent to which the NHP model matches human responses and how easy or useful you think it would be in the future to generate a human testicular model for translation. So I think that addresses that. The other question that we had from Anthony was, I assume Zika virus affects female reproductive organs also. If so, do you or others have intentions of developing MPS models to study this, or do these exist already? Any plans to develop human models?
Yes, we are also developing in my lab, along with another scientist at CBER, human placental barrier models, because Zika virus, in addition to being sexually transmitted, where it can infect the developing fetus through ascending infection into the uterus, can also cross the placental barrier. So we're developing similar models using the tissue chips with human placental cell lines, such as the BWU, syncytiotrophoblasts, placental fibroblasts, and endothelial cells as a barrier, and monitoring Zika virus migration through that barrier, as well as looking at the migration of, or the transcytosis of neutralizing antibodies to the Zika virus, which could also have a negative impact on the developing fetal tissue.
It's super interesting. It seems like it would be, there's really neat potential there for integrating that type of a barrier model into that system where you're looking at infection into the neurosphere as well, since that's, to my understanding, the most sensitive part of the developing fetus to the viral infection. Great. Okay. Thank you so much. In the interest of time, we're going to move on. So our next presentation is from Mark Williams at NIAID on moving beyond SARS-CoV-2, NIAID efforts for pandemic preparedness and prototype pathogens.
Thanks, Nicole. Next slide, please. So it's nice to have Dayton just before us to give us this sort of idea that, you know, besides the COVID epidemic that we've been struggling with for many years now, you know, there's a lot of different outbreaks historically that we within NIAID and DMID, the Division of Microbiology and Infectious Diseases, have dealt with the filoviruses and other coronaviruses, MERS and SARS-1. What I'll talk to you about, at least in the first few slides, is our efforts, you know, to look forward, you know, in terms of, you know, how can we prepare ourselves for what's coming down the pike. So next slide. So just kind of, you know, thinking about lessons that we've learned in terms of COVID, the years of study and history that, virology, immunology, facilitated rapid and successful vaccine design. The messenger, obviously the messenger RNA platform was key to this. But if it was a different viral family that wasn't quite so tractable to these things, we might have been a different story. So we really need to think about how to prepare ourselves moving forward. And critically, I think one thing I've heard echoed many times at many of these meetings is this interagency cooperation. I say here U.S. government, but clearly across pharma, across all different agencies worldwide, it was critical to successes, and overcoming obstacles as we were going along in terms of product development. So next slide. So thinking forward, that this preparedness that we enabled successful or shortened timelines for COVID responses. NIAID has come together and developed what we call pandemic preparedness plan. And so the idea is to really sort of systematically characterize known pathogens, and before they can emerge, so we have a bank of knowledge or a bank of products in hand. With the goal really of shortening timelines between onset of outbreaks and filling in gaps between research and infrastructure, technology, knowledge, all of these things that are critical, diagnostics, all these different tools that we need to have in place when the next outbreak happens. And again, synergizing all of our efforts across all the government agencies and key partners and stakeholders. So that's the sort of view. And then so the question is, how do you go about this with attacking something that's an unknown going forward? So next slide, please. And so the way that we decided to work at this is to have what we call the prototype pathogen approach. And so amongst all of the different virus groups and families, we, with the scientific community and other stakeholders, identified individual pathogens, representative of the different groups and families that we could then focus efforts upon in terms of understanding pathogenesis, assay development, biology, preclinical models, all of those different steps that you need to, be able to understand and develop, medical countermeasures for these different viruses, and not try to, approach the whole different, number of viruses that are out there, but hopefully that would allow this sort of, focused and structured efforts to be able to get us to the point where we would have monoclonal antibodies or vaccines that would be, platforms that would be tested and ready to go when the next virus that, that could be adapted when the next virus came around the pike. So next slide. So as I said, through meetings with the different, partners, and global stakeholders, we identified different virus families shown on the left of these different boxes, the arenaviruses, hantaviruses, et cetera, and then identified prototype viruses within each of these families that we would then focus efforts upon with our grants and our contracts within NIAID, the financial support, to try to be able to improve the different levels of preclinical work and get us to the point where we had assays and understandings to be able to move quickly in the case of a coming outbreak. And I'm not going to go through all the different viruses, but hopefully you'll see some that are familiar to you or maybe not so familiar, to stimulate some interest moving down the line. And so this is sort of the approach that we've taken in our group, is setting up existing contracts to be able to establish animal models and preclinical development to be able to understand and test candidate therapeutics and vaccines against a variety of these different prototype viruses. So next slide. So this slide sort of details a little bit some of the existing funding that NIAID has funding vehicles that NIAID is using right now to try to advance these efforts with the prototype viruses, the revamp centers for the vaccines and monoclonal antibodies, APP, the antiviral program for pandemics, which is more for smaller molecules and medical countermeasures, and then the creeds, the more of an epidemiologic focus on being able to understand and monitor, worldwide for viruses of pandemic potential. So, but what I'd like to do within the next few slides after this is sort of a public service announcement and focus on some of the core programs that are our group within DMID and services that we supply or provide to the scientific community that we feel will also help advance and complement these grant efforts. And these are contract vehicles by which we engage contractors to provide services to outside investigators, and I'll explain more about that in the coming slides. So next slide. So the first one of these services that I'll focus on is BEI resources. So hopefully you're familiar with this. This is a contract that DMID supports. And the mission is to provide reagents and tools and information for investigators studying priority pathogens. And we've now pivoted and moved along the way to new emerging infectious diseases, vectors of disease, and even non-pathogenic microbes. And you'll see in the bullets on the bottom, this is sort of a very broad range of reagents and organisms that are provided, and I'll emphasize those last three words there in green on the mission above, that these are all provided free of charge to the investigators. You just register for the BEI resources. We make the sort of analogy that it's almost like Amazon in one step shopping, where you can pick and choose different antigens or different antibodies, different viruses, different pathogens of interest, and then you just have to pay shipping, and the costs of the materials are free to the investigators. So we feel like this is a really valuable resource for the community, and encourage everyone to go and check it out at the website there listed at the top of the slide. And it's really been a valuable tool for NIAID to be able to pivot from one outbreak to the next. So we helped support diagnostics when Zika was outbreaks. So Zika in pregnancy, designing diagnostic assays for Zika, and clearly it's been just a outstanding tool for SARS-CoV-2. And so I want to highlight that in the next slide. And so one of the real values I felt in terms of our efforts in the COVID pandemic was the production and supply of what we call well-characterized challenge material. And I think this is really important in the context of one of the subjects of this talk or this meeting, is that we need to think about, in terms of regulatory acceptance, the things that are, FDA or the regulatory bodies are going to be looking for. And one of the things that are going to really be important in terms of a package is that the pathogens that you're using are well characterized and consistent between investigators and between experiments. And as many of you have found, those of you that have worked with the SARS-CoV-2, this proved to be very difficult, because when you started culturing SARS-CoV-2 to make virus for your experiments, what we found was that there was a number of single nucleotide polymorphisms and even deletions in the genomes of these viruses. And this really impacted the infectivity of these viruses. And just the data that are shown there on the left graph is the infectivity in hamsters with a well-controlled virus stock, and the one on the right is the infectivity when a strain that had been cultured in vitro and developed some deletions in the key furin site for that mediates infectivity. And you can see hopefully the large decrease in infectivity in these in vivo models. So we took on the challenge of basically creating large volumes of harmonized, well-characterized challenge material for SARS-CoV-2. To date, we've covered over 45 unique variants of concern that are all available free of charge through BEI. And there's, as I said, large stocks such that you don't have to grow the virus yourself. You can just go to BEI, buy the virus. They're provided in low volume, in single-use tubes, and use the virus in your infection, and then have use new tubes for each subsequent experiment, and be confident that you have a harmonized virus across all of your studies. We do a lot of work in terms of optimization of growth cells, MOIs, utilizing next-gen sequencing and annotation to ensure that we have really well-characterized virus stocks for all of these unique variants. And so I'm bringing this up because, part of this with the pandemic preparedness efforts, now we're transitioning to apply these same sort of techniques and approaches to a lot of the prototype pathogens that I talked about in the earlier slides. So we have in hand right now stocks of Nipah and MERS. In process, we're making stocks of Lassa, Ebola. So working with the scientific community and funding agencies, identifying key strains, important strains of each of these different viruses, growing up leaders and leaders of the well-characterized challenge material that, again, will be available free of charge through the BEI resources. Next slide. So the other service that we supply to the community is preclinical services. And so again, these are a set of contracts that respond to external requesters and help fill key regulatory gaps for people putting together, regulatory packages and advancing development of particular countermeasures, whether they be therapeutics or whether they be vaccines. And we provide what we call gap filling services throughout a variety of different steps, in vitro manufacturing, even GMP manufacturing, immunogenicity of vaccines, and importantly for this meeting, the research resources or preclinical models of infectious diseases. So next slide. And so these preclinical services provide a forum or a service by which people that are developing vaccines or therapeutics can come to us, and we will take their product, hand it over to our contractors, maintaining confidentiality and intellectual property. So there's no loss of anything like that. But basically, for free of charge, we will provide data for, as I said, PK, tox, manufacturing, infectivity, all wide variety of different services to help move along promising countermeasures and vaccines in a wide variety of pathogens and concerns. So next slide. So this is, I think, really valuable and underutilized, in my opinion, which is why I'm spending some time to talk about it today. And it's actually available to anyone out there. I mean, you don't have to have NIH funding. You don't have to be in academia. We've supported nonprofits, small and large pharma, even other government institutions. It can be domestic or foreign institutions, and there's a simplified request process. Obviously, we're not going to support everything, but we evaluate and prioritize the things that we're going to try to help support. In terms of the significance, the innovation, and the readiness and technical readiness sort of stage, the product development plan that the product developers have. Next slide. So. And the reason I talk about these preclinical services at this sort of meeting is, one, I think it's valuable, but I think it's also a place where we are really trying as an organization to implement microphysiologic systems as a tool for product development. And so one of the besides the increasing potential acceptance and within regulatory agencies, we think that there's some key values in terms of implementing and utilizing microphysiologic systems to support product development, and that's to evaluate, as we've talked about, in a relevant cell, a relevant tissue or cell system, minimize the requirement for animal adapted pathogens or modified hosts, like we've talked, needed to use for some of the, you heard about, Dayton talk about for Zika. We had to do that for coronaviruses. You can evaluate host directed medical countermeasures, countermeasures directed towards, human targets, human proteins. Human biomarkers of disease. Tyler talked about different biomarkers that you can measure out of these systems. And obviously, the driving the views of the 3 R's, reduce the reliance upon experimental animals. So next slide. So within our preclinical services, we currently have contracts utilizing 2 different microphysiologic systems that kind of span the range of technology. One being human intestinal enteroids, run by a group at Baylor, or led by a group at Baylor, which was used as a model for human norovirus. And so this system has been well characterized in terms of supporting norovirus growth, and we've used it to evaluate a range of different medical countermeasures and readout of virus proliferation in these enteroids. And the other system is a much more complex system, a human lung microphysiologic system, run through University of Texas Medical Branch as our contractor. And this is a reconstituted scaffold. So it's not a classic organ on a chip system, but it's reconstituted with the alveolar cells, fibroblasts, smooth muscle cells. We can form it with or without tissue macrophages, and run it with or without a vascular component with flow. We've used it to demonstrate productive infection. We actually started using it before the pandemic, looking at avian flu and Francisella tularensis as models for being able to use this system in BSL-3. And by luck for us, I don't know about luck, but then the pandemic comes along, and now we could easily pivot to SARS-CoV-2 work. And we've been able to use this to test the efficacy of a number of different countermeasures and candidate therapeutics that I'll illustrate in just one data slide. Next slide. And so this is utilizing the human lung MPS, looking at replication across the 3 different graphs of Washington 1. And then as time moved on, we also looked at some of the other variants of concern. This is the initial Omicron, the BA1. And this is just showing you data showing the inhibition or lack thereof by a variety of different medical countermeasures that we've screened in this system. So this is measuring viable virus by TCID50 viral titers, and you could see a large increase in virus, and then over time a decrease, which is associated with loss of viability of the human airway epithelial cells. In the open circles, and in the closed squares for on the far left, the EIDD 1931, that's molnupiravir. You can see a couple of log decrease in the viral load with exposure to that drug. Camostat in the middle panel is a protease inhibitor, shows a much more potent inhibition of viral replication. And then, as you might expect, dexamethasone didn't show any inhibition of viral replication in this system. We're looking at at different downstream biomarkers in these systems as well. And so this is just an example of how we were using these sorts of systems to be able to advance medical countermeasure study. So next slide, and the last slide. So I just wanted to put this out here as a sort of, how do we use these things? If we're going to use these sorts of systems to support medical countermeasures, Dr. Ishida, identified a lot of these sorts of concerns in terms of, thinking about how we're going to get regulatory submission and the idea of qualification of these systems. We, in our group, we've been really involved with the animal model qualification system through the FDA, and we have To date, the first and only non-human primate animal model qualified under the drug development tools with FDA. And we put together this slide to sort of illustrate, and I should say the hyperlink on the bottom is the link where you can see the context of use for this model that we had. But put together this slide to sort of illustrate the challenges and the things that we need to be thinking about in terms of translating the animal model qualification views now to thinking about MPS. So the well-characterized pathogen, making sure it's very well characterized in terms of sequence and morphology. The challenge process, you heard about aerosol and different pathways of ways of challenging and introducing the virus or pathogen into these microphysiologic systems. We think about the well-documented animal model, think about the cell types. I was really heartened to see, Professor Ishida talk about, what sort of cells, what sort of markers do each of these cells have? Are we controlling that from experiment to experiment and across all the different users and model systems? And obviously then the pathophysiology. Is this model representative of human disease, and what sort of outcomes and biomarkers are we able to use to evaluate the use and productivity of these systems and their ability to support regulatory submissions down the line. So hopefully that will provide some things for discussion later on in the process, but I'll stop there. I'm happy to take any questions. Thank you.
Thank you so much, Mark. Really tremendous resources for the community. I think you're maybe doing yourself a disservice to compare yourself to Amazon. I think you guys are better than Amazon. All right, so we are pushing up against our break. So I do want to give everybody a few minutes to get up and stretch their legs and have a bio break. But I want to pause and see if there's any clarifying questions at the moment. I'm not seeing any in the chat. So let's go ahead and take a break. We'll take an 8-minute break just to stay on time. And there is a question now for Mark in the chat about, does the NIAID program have capacity for new antibacterials? So maybe, Mark, if you could humor me and type the answer in the chat,
sure,
so that
absolutely.
everybody can see it while folks are on break. And so we will reconvene at 10:35. Thanks, everyone. Okay, everyone. So I hope you all took a few minutes to get up and stretch your legs, take a small break. And now we're going to restart our exciting agenda. So thanks, Mark, for answering the question in the chat, really informative. So now we're going to move on to Gwen Fuel's presentation from Synvivo. She's going to be speaking to us about MPS to model and evaluate immune endothelial cell interactions in inflammation and sepsis. Gwen, over to you.
Thanks, Nicole, and thanks for the invitation to present in this workshop. It's been really fascinating listening to all of the presenters already. As Nicole mentioned, I will discuss how the Synvivo MPS platform has been used to model and evaluate immune endothelial, neutrophil endothelial interactions during inflammation and sepsis. Next slide, please. This is just a brief introduction on the Synvivo MPS platform. So on the bottom, you can see some of the Synvivo microfluidic chips. We have either actual microvascular networks that have been modeled from in vivo scans and include the complex geometry of in vivo vasculature and variable shear, as well as some idealized designs on chip. All of our co-culture chips have vascular and tissue channels that are actually separated by pre-engineered barriers. And you can select these barriers depending on the unique needs of the tissue or the organ that you're trying to model. It's a side-by-side architecture, so you can also develop complex co-cultures, still maintaining the real-time visualization and quantitation because they all stay in the same focal plane. And of course, the models incorporate physiological flow and shear stress Another point I wanted to call out here is that each channel is independently addressable, so it allows both on-chip and off-chip analysis. And so we've used this MPS platform and developed several models for that are available as products or contract services. These are our blood-brain barrier models, several vascularized tumor models, a lung air-liquid interface, tox models, including vascular toxicity. But today I'm going to focus on our inflammation model and our assays, and especially describe how they've been used to study sepsis. Next slide, please. Again, a quick introduction. So sepsis is defined as a life-threatening organ dysfunction caused by a dysregulated host response to infection. So it's a very debilitating disease and starts with infection or injury, eventually leading to systemic inflammation, followed by vascular endothelial damage, increased permeability, tissue injury, and multiple organ failure, ultimately. In the US alone, there are greater than 1.7 million cases per year, with a mortality rate in excess of 270,000 deaths per year. It's one of the leading causes of death in US hospitals and costs the healthcare system over $24 billion a year. Next slide, please. The current treatments for sepsis are antibiotics and supportive therapies like mechanical ventilation or fluid management. There are no specific pharmacological therapies for sepsis-induced tissue injury. In fact, all recent sepsis drugs have failed in clinical trials. Now, the neutrophil endothelial cell interactions that result in increased neutrophil influx into organs and cause endothelial barrier disruption, they play a critical role in the development of sepsis-induced organ damage. And we are modeling this in our MPS inflammation assays. Next slide, please. Here's another piece that I wanted to talk about. So if you look at activation of endothelial cells in sepsis versus normal healthy cells. So in the normal healthy tissue, the glycocalyx coats all the healthy vascular endothelia. And this is a layer that's critically involved in maintaining vascular integrity, allowing neutrophils and other white blood cells to pass through the microvasculature without much interaction with endothelial cells. However, when you have an activating agent like bacteria or sepsis or radiation even, the vascular barrier is impaired because of glycocalyx breakdown, endothelial cell apoptosis, and the junction protein dysregulation. So you get chemoattractants and chemokines released by the damaged tissue. Adhesion molecules are also activated on these endothelial cells. So then through a combination of fluidic and molecular forces, you have leukocytes or neutrophils start rolling, adhering to the endothelial cells, and then migrate into tissues, and they start releasing cytokines, reactive oxygen species, and proteases to fight against infection. And typically the process helps the tissue unless it's out of control, like in sepsis. The resulting inflammation will lead to progressive tissue damage, finally causing the multi-organ failures. And so again, we are modeling endothelial cell activation and neutrophil adhesion and migration in our MPS models. Next slide, please. So here's what, here's some images that shows some of the microvascular network designs that we're using to model inflammation. So on the top left, you can actually see the chip with microvascular network geometry, and it's been reproduced from microvascular networks actually observed in vivo. You can see in the middle here, you have a close-up in this image shows a close-up of the vascular channels and the tissue compartments. And these are separated by a barrier that allows communication between the vascular and the tissue sides. You can grow all sorts of endothelial cells in the vascular channels. And when you put endothelial cells under flow, they form these complete lumens. And through which you can deliver, your drugs, immune cells in this case. They can be introduced under flow and shear conditions. The shear map on the bottom here shows variations in shear rates and flow conditions in these vascular networks and on bifurcations on the chip. And blue indicates low flow rate and red indicates a high shear rate. So the effects of shear flow and the vessel geometry on neutrophil adhesion and migration can also be determined in this model and assay. Next slide, please. So here, these are some videos showing the functionalized MPS assay in action. And on the top, actually, the top is a video as well. If you can click on that, top right, you can see the formation of the complete lumen with endothelial cells when you put it under flow. This vascular lumen can support flow and the introduction of leukocytes into the endothelium, both in the microvascular networks, which you see on the right bottom side, as well as in more idealized designs on the left side here. You can see in the microvascular networks that the flow rates in some of the vessels are slower and some of them are faster. Can you click on that? The video right there. Yeah, I don't know if it's on. So the flow rates in these vessels, some are slower, some are faster, which is very consistent with what you'll see in vivo. So if you want to study leukocyte endothelial interactions during inflammation, you can set up your model with endothelial cells seated in the vascular channels. They get put under flow, they form the lumens, and then you can activate these endothelial cells using TNF-alpha or other inflammatory signals. You can introduce a chemoattractant like FMLP in the tissue chamber. And then when you inject your labeled leukocytes or neutrophils through the inflamed endothelium, you can watch them interact with the endothelial cells under physiological shear conditions. Some of them will adhere to the endothelium, which starts the process of extravasation across from the vascular side to the tissue side in response to the chemoattractant. So this assay essentially reproduces the entire leukocyte adhesion cascade in a physiologically realistic 3D environment. So it encompasses circulation, rolling, adhesion, migration of leukocytes into the extravascular tissue space under physiologically relevant flow conditions on the chip. And so you can visualize all of these processes happening in real time, but you can also quantitate them. So next slide, please. So next slide. There you go. So you can quantitate rolling, adhesion, and migration of neutrophils on the chip. And here's an example of migration of neutrophils. And these neutrophils have been labeled with a fluorescent dye into the tissue compartment after 120 minutes of continuous flow over the endothelial cells that have been inflamed with TNF-alpha. So the solid arrows on the top right panels show a rolling neutrophil. These are panels 1 and 2, which become adherent in the panel 3. The dotted arrows in the top right panel show firmly adhered neutrophils. And then the bottom right show neutrophils migrating from the vascular channel through the barrier into the tissue compartment over time. And so you can take images over time or video over the course of the assay, and then you can analyze it to count the numbers of rolling, adherent, or migrating neutrophils, and importantly, how they can change when inflamed or when they are treated with anti-inflammatory drugs. Next slide, please. So I'm going to walk through some data here, just comparing human and mouse MPS models. We know that a lot of drugs that have been developed in animal models don't translate into the clinic, but with MPS models, you can compare human and mouse side by side. And here's some data from a study that was published by our collaborators at Temple University, Mohammad Kiani and Laurie Kilpatrick labs. And they looked at human and mouse models and compared their responses to inflammation caused by TNF-alpha activation. So in the presence of TNF-alpha, as well as in the presence of an inhibitor to PKC delta, which is an important regulator of the inflammatory response. So you can see here that over time, over 60 minutes, we are looking at neutrophil migration, you see that after TNF alpha treatment, there's neutrophils are migrating towards the FMLP chemoattractant signal. And you can block that migration by using the PKC delta inhibitor. However, if you look on the right side, the murine neutrophils treated with the inhibitor to PKC delta continue to show increased migration in response to the chemokine. So this is kind of an important finding because the PKC delta inhibitor was not effective in the mouse model, but was effective in a human model. And so this is data that could illustrate the real situation where an inhibitor that's very effective against human cells may be abandoned because it was ineffective in the mouse study. So this is an opportunity to use the MPS systems to compare human and mouse models side by side, and it's quite powerful. And so these MPS models and assays could be used as a tool to assess anti-inflammatory drug candidates and increase their clinical translatability. Next slide, please. So the same study also looked at adhesion molecule expression under shear flow conditions in the MPS model and saw that the pattern of expression is also different of one of these adhesion molecules. The ICAM1 is different in human and mouse cells. So in particular, ICAM1 is upregulated in human endothelial cells, but not in mouse endothelial cells. Again, this side-by-side comparison is important because if you were developing an inhibitor that's effective against ICAM1 adhesion molecules, the inhibitory effects may not be detected if you were to use a mouse model. So using human cells and tissues, we see here again that the PKC delta inhibitor was effective at reducing the expression of ICAM1 to the same levels as the no treatment controls in the human cells. Next slide, please. So we have also modeled neuroinflammation in the MPS platform. And neuroinflammation often develops in sepsis that leads to activation of the cerebral endothelium, causing increased permeability of the blood-brain barrier, as well as neutrophil infiltration. So we can model this using brain microvascular endothelial cells and astrocytes or astrocyte conditioned media. And this is a BBB model that is functionalized with brain endothelial cells showing complete lumen formation under flow. Next slide, please. This data is showing the human BBB model functionalized with brain endothelial cells and astrocyte conditioned media that was treated with TNF alpha to activate the endothelium and showing increases in vascular permeability as well as tear after activation. And then you can also see that using an inhibitor caused a protective effect against this TNF alpha induced inflammation and vascular permeability. So in this case, permeability was measured using FITC-dextran, and then tear measurements were made on chip in our tear-enabled blood-brain barrier chip. Next slide, please. You can also see that PKC delta inhibition reduces neutrophil adhesion as well as migration across the inflamed blood-brain barrier. So you can see the control and TNF alpha showing the number of adhered neutrophils increases when the brain endothelial cells are inflamed, and you can knock that down with the PKC delta inhibitor. A time-dependent increase is also seen on the right here in the number of migrated neutrophils across the TNF alpha activated endothelium in response to the chemoattractant FMLP. So the neutrophil migration across the cytokine activated endothelial cells was almost completely inhibited after the PKC delta inhibitor and became even significantly lower than the control level of the neutrophil migration. So you can see that we can model activated endothelium not only, also across the blood-brain barrier. So I want to spend the last few slides, next slide, please. I want to briefly mention some new and exciting work from our collaborators at Temple University using the MPS model. So Doctors Kilpatrick and Kiani undertook a clinical study that evaluated neutrophils from sepsis patients compared to healthy controls. And they were actually able to identify neutrophil phenotypes that also correlated with disease severity and patient outcomes. The work was submitted for publication, so I'm just giving a little teaser on some of the data that was generated using the inflammation model. So in this study, 9 healthy adult donors and 34 patients who were admitted to Temple University ICU for sepsis were recruited. And information on clinical lab tests, as well as data around whether or not they were on ventilators, were collected. Their neutrophils were isolated and tested in the in vivo MPS inflammation model. Next slide, please. So these neutrophils isolated from healthy donors and sepsis patients were tested for their response to a cytokine cocktail. So cytomix in this slide, which is a combination of TNF alpha, IL-1 beta, and interferon gamma. And so it was compared against buffer-treated cells. And so the effect of endothelial cell activation was measured using neutrophil adhesion to human lung microvascular endothelial cells. And we watched the adhering in the vascular compartment and migration into the tissue compartment. And as you can see on this slide, there were distinct differences in total neutrophil adhesion among these sepsis patients in the way they responded to the cytomix activation. If you looked at total neutrophil adherence, the hypoimmune group of sepsis patients did not respond to cytomix activation, and they were not significantly different from buffer controlled. So a clear, phenotype we see with total neutrophil adhesion. Now, they had done discriminate analysis of the distribution of neutrophil functional adherence at different shear rates and migration patterns to identify these 3 patient groups corresponding to the 3 distinct clusters of neutrophil responses to cytomix. And so you see here that it's a hypoimmune, a hyperimmune, or a hybrid phenotype, where the hypoimmune does not have a lot of adhesion versus the hyperimmune or the hybrid. Next slide, please. Similarly, when you look at neutrophil migration across the brain microvascular or the lung microvascular endothelial cells in sepsis patients, you also saw distinct patterns in response to cytomix activation, where your hyperimmune group showed increased migration, but the hypo or the hybrid group had not very many migrating neutrophils compared to the control. So next slide, please. Next slide. So there's 3 functional neutrophil phenotypes. So you have your hyperimmune, which has increased neutrophil adherence to the lung microvascular endothelial cells, increased neutrophil trafficking, and they correlated with disease severity such as pulmonary function, mechanical ventilation, and the length of stay. So these patients had increased systemic neutrophils, increased mechanical ventilation requirements, and increase in the length of stay in the ICU. The hypoimmune phenotype that had reduced neutrophil adhesion and migration also correlated with reduced requirement for mechanical ventilation and decreased length of stay in the ICU. And then you had your hybrid phenotype where you had increased neutrophil adhesion, but lower neutrophil trafficking. And that also, it did have a mixed phenotype where they did increase the number of patients requiring mechanical ventilation, but they spent less time in the ICU. But this kind of data highlights the power of the MPS systems to provide relevant in vitro data that can be predictive of patient outcomes, in this case, like the need for mechanical ventilation. It can also help distinguish sepsis patients who would benefit from specific treatments, because we know that there's no real drugs to treat sepsis patients. They are largely on supportive care. So it does provide a path towards personalized strategies and improved patient care. The other thing, the identification of these distinct phenotypes in the sepsis patients may also provide some insight into the failure of sepsis drugs in clinical trials. Next slide, please. So in summary, the organ-on-chip platform can be used to model inflammation and evaluate endothelial cell activation across different tissues and organs. And I showed you, inflammation models with both the lung microvascular endothelial cells, as well as the blood-brain barrier, brain endothelial cells. It can be used to screen potential anti-inflammatory therapeutics, and you can compare human and animal species to identify differences that can impact drug development. Lastly, the identification of sepsis patients with diverse functional neutrophil responses can also help distinguish patients who might benefit from specific treatments. Next slide. So I just want to again acknowledge our collaborators and thank them for the collaboration, and also for providing me some of their slides for the clinical data, which is submitted for publication. So the inflammation models from Synvivo are available as products, kits and chips, as well as contract research services conducted at our facility at the Hudson Alpha Institute for Biotechnology in Huntsville, Alabama. So thank you very much for your attention, and here's contact information.
Thank you, Gwen. Excellent. One very quick question for me. So my understanding is that there is a FDA-approved drug, Giapreza, that's a synthetic human angiotensin II that's approved for treating sepsis. And obviously, I think the predominant mechanism is an effect on blood pressure, but there's some data that shows that it has an immunomodulatory effect as well. Have you thought about testing that in your system, particularly with the sepsis patient-derived neutrophils with varying phenotypes?
So this is a great question, and our collaborators at Temple University are conducting these studies with sepsis patients, and this is something that is once we have this initial publication, I'm sure there will be follow-up studies like this.
Okay, great. Thank you. There is also a question in the chat that if you wouldn't mind typing the answer to that would be great.
Okay.
And so now we're going to move to a presentation from Don Ingber. I saw him online previously, but I think the intention was he had recorded his presentation, and then hopefully he'll be able to log back on later for the panel discussion. So John, do we have the presentation recording ready to go?
I am here if it's not working, but it's short, so it might save time. Thank you for giving me this opportunity to share with you what we've done over the past 15 years on human organ chips, and particularly focusing today on human organ chip models of infectious disease. The chips that we use are two-channel chips. They have two channels separated by a porous membrane that we coat with extracellular matrix, and we could put different tissue types on either side to recreate tissue-tissue interfaces, usually epithelial and endothelial cells, but also we can put stromal cells there as well. And we have side chambers that have cyclic vacuum applied that can replicate mechanical deformations like breathing in lung and peristalsis in the intestine. And of course, we have fluid flow individually controlled in each channel, or we could have air in one and fluid in the other. This is now made available internationally. You can buy them from Emulate, which I'll describe at the end. Our first breakthrough was 2010, where we modeled a human lung alveolus or air sac. And I'm not going to go into all the results, but that really provided the proof of principle in a series of studies that we can model human diseases and toxicity, drug toxicities, drug efficacies, identify therapeutic targets, facilitate drug advancement through clinical trials and gene therapy delivery. And it's won numerous honors. But we've now expanded that to over probably 15 different organ chips. And we have definitely pursued infection models. And so first example, and I'll give you a few, is the human lung airway chip, which we showed lined by bronchial epithelium and interface with lung endothelium, that we recreate ciliary differentiation, directional motion, mucociliary transport. And we had funding from Cystic Fibrosis Foundation. They provided us with cells from patients with cystic fibrosis. And when we took their bronchial epithelium on chip, the first thing we noticed is that they have, just like in patients, increased ciliary differentiation, shown at the top right in green. They mimicked, beating frequency, mucus production, baseline immune state, cytokine release. But for this talk, we modeled Pseudomonas aeruginosa infection because this is present a bacterium in all of our lungs. It's normally homeostatically controlled, but in patients with cystic fibrosis, there's overgrowth, and this is a major cause of morbidity and mortality. And interestingly, we can model that beautifully on chip, as shown at the bottom right, with much higher abundance of bacteria in the CF chip under identical culture conditions. About this time, DARPA and NIH, 2017, asked us to see if we could model potential pandemic respiratory virus infection, but at that time, the focus was on influenza. And at bottom right, you can see H1N1 influenza virus labeled with GFP infecting in real time. These are optically clear chips. You could do any imaging. You could do in vitro and in vivo on them, so that adds huge power to them. But you could also see that we can model cell and tissue responses to viral infection here, loss of cell junction, cilia, even endothelial damage. And then we could model different viral strains and replicate the viral strain dependent virulence seen in patients based on viral load. Importantly, we can model comorbidity effects. In this case, we made chips with patients from chronic obstructive pulmonary disease, COPD, 10-fold higher viral load with the same infection. And this is important because this is often missed in animal studies, but basically comes back to haunt you in clinical trials. We could model the host response to infection in terms of cytokine release or cytokine storm. Again, 6 different cytokines, the levels of release matches what's seen in patients. Immune cell recruitment, we could circulate human primary PBMCs through the vascular channel, watch them cross both tissue layers, cell layers, and tissue interface when viral infection takes place, and then that clears the virally infected cells. We could also model drug effects. This is also Tamiflu, also known as Tamiflu. Interestingly, not only do you see suppression of viral infection, but you see the same time dependence. It's only active in the first 2 days after infection, and that's exactly what is seen in patients, and FDA has only approved it for that. We also could model evolution of, or spontaneous evolution of variants of concern, if you like, of virus. We take our airway chips, infect with influenza, treat with drug at 90% level for a few days, and then take a droplet of fluid medium through the airspace and go to the airspace of another chip treated the same way, and repeatedly do this until we see evolution of viruses that are completely resistant to, in this case, amantadine after 8 passages. When we genome sequenced, we found all of the known mutations seen in patients who became resistant to this drug, as well as a few no one's ever seen before, meaning we may be able to use this to identify variants of concern that are going to emerge in the future. This is when COVID-19 hit. 2 postdocs in my lab who were from China. They started to test drugs very quickly against the pseudotype virus they engineered. And they chose drugs that were previously shown to be active against the original SARS or MERS related coronaviruses. They found 8 drugs, including hydroxychloroquine and chloroquine in January 2020 that were active in the low micromolar range. But when we tested these drugs in the airway chip, and now we flow them through the vascular channel at a Cmax, a clinically relevant dose, hydroxychloroquine and chloroquine had no effect, whereas a related anti-malarial imidacloprid was effective. We confirmed this in a hamster SARS-CoV-2 and BSL-3 lab model, and that drug moved to clinical trials across 20 sites in Africa. More recently, we used the breathing alveolus chip, and we found that breathing activates innate immunity, stimulates type 1 interferon release, which is protective against viral infection, and that resulted in suppression of viral infection in terms of reducing viral load, decreasing cytokines on chip. And we worked out the mechanism on chip using RNA-seq and other methods, and we identified the RAGE, the receptor for advanced glycation end products, as a major mediator of this response. And we identified a drug that inhibits RAGE called aziliragon, and we completely prevent cytokine storm in this model. And these data were actually included in an IND submitted to the FDA by a company called Cantex to move this drug to clinical trials for COVID-19. We've even discovered entirely new therapeutics. In this case, we did a siRNA screen to look for host response genes that may protect against influenza infection. We found a family of short duplex RNAs that it turns out it wasn't siRNA suppressing gene expression. It was that they all had the same structure, about 20 base pairs, very unique structure, and they were potent inducers of type 1 and 3 interferons, again, protective. And you can see here interferon beta, 30 and 15-fold increases in human alveolar and airway epithelium. And when we treated our chips with airway chips with these viral infection with H1N1 influenza, H3N2 influenza, potent inhibition. In vitro, we inhibited SARS-CoV-2, MERS, and common cold coronavirus as well. And then we did it in a BSL-3 lab, a SARS-CoV-2 infection model in mouse, and got 5 log reduction in viral load. So this is now a broad spectrum antiviral inhibitor that was discovered leveraging organ chip technology. We've also done many other chips. So that we developed human intestine chip years ago. Initially, we call it a gut chip lined by human CACO2 endothelial cells and endothelium. But more recently, we do primary chips with cells from patient-derived intestinal organoids, which we expand in 3D gels, but then break up, put in the top channel of the chip and endothelium in the bottom. Both form beautiful intestinal villi. And we initially used the gut chip to model enteric virus infection, in this case, Coxsackie B. And you can see at the right, you can see over 24 to 48 hours, you get cytopathic zones. You could see polarized release of virions from the apical domain of the epithelium, as well as polarized release of cytokines. And now, as I said, we use organoid-derived chips, and just showing here that when we did transcriptomic analysis, we actually found that the intestine chip made from duodenal organoids was closer to the duodenum than the organoids themselves, which were pretty good to begin with. But now, by creating oxygen gradients on these chips, we're able to co-culture complex human microbiome, in this case, over 200 different types of bacteria from 11 different genera, similar complexity to stool on these chips, while keeping the intestinal epithelium alive and happy. And this is a major advantage over organoids in cell culture, where they can only do it for about 24 hours before they die. And using this, we modeled a disease called environmental enteric dysfunction, EED, with support from the Gates Foundation. This has contributors of malnutrition. It's basically malnutrition in low resource nations, but also has pathogens and dysbiosis contributors. And just to cut the long story short, we were able to show that we could model this disease by exposing chips lined by cells from kids with EED in Pakistan. We get organoids from them with malnutrition. But now if we add a pathological microbiome from these kids, so it's sort of an infectious microbiome, if you like, you can see greatly increased inflammatory cytokines with the EED microbiome at the right. And now you can see by combining malnutrition minus N minus T with this GD as a consortium that Jeff Gordon helped us get from kids, you can see increased intestinal barrier disruption as well. And then last year, Gates asked us to model bacterial vaginosis on chips. So we had to create a vagina chip. This is primary human vaginal epithelium now interface with stroma fibroblasts to get an epithelial stromal interface. We get beautiful epithelial differentiation and stratification. And the reason they did this is they wanted to move a live biotherapeutic product. So basically a consortium of healthy bacteria, in this case dominated by L. crispatus, that come from healthy vaginal microbiome that have been purported to protect against bad microbiome that's dominated by Gardnerella vaginalis. And they want to move this to a clinical trial. In fact, they are this year in South Africa. And so we've been developing this as a test bed for these live biotherapeutic products. And here I'm just showing you one example where the black bar is inflammatory cytokines in these chips, which are very high when you infect with the bad bug, G. vaginalis. And these are 3 different formulations of L. crispatus consortia in live biotherapeutic products. And you can see differing responses, but 2 out of 3 give you potent suppression of inflammation. And these 2 are the ones moving to clinical trials this year. And then finally, we have developed a human lymphoid follicle chip that is a fantastic model for vaccination responses. This is B and T cells from peripheral blood from apheresis collars at high density in matrigel in one channel, we superfused from the other. And if you take commercial flu zone vaccine, influenza vaccine, you get spontaneous organization of germinal center-like structures, plasma cell formation, antibody class switching. If you put dendritic cells in this as well, you get high affinity antibody against the hemagglutinin antigen in the vaccine. And we could even measure cytokines in the effluent, and they look just like what's seen in patients who've been vaccinated. And we now have been using this to model adjuvants and other vaccines. But very recently, it's unpublished, we've modeled infection with Borrelia burgdorferi, which is Lyme disease infection. And amazingly, we see what is observed in patients, which is suppressed T cell activation and hypergammaglobulinemia with IgA, IgE, which we have never seen in 80 patients in our vaccination studies. And it varies patient to patient, which may explain why some people get chronic Lyme. But this is the beginning of infection-induced autoimmunity modeling using organ chip. So the take-home message is that these 2-channel organ chips are excellent models for infectious disease. They're outstanding animal replacements. They're also mechanistic drug discovery tools that can provide new insight into pathophysiology, be combined with advanced analytical approaches, enable rapid drug repurposing and accelerate drug discovery of novel therapeutics. And I have to explain that I'm a member of the board, chair the SAB, and hold equity in Emulate, which now sells these instruments and chips around the world, and invite you to the website if you'd like to learn more. And I thank you for the time.
Thank you so much, Don. As usual, incredibly impressive whirlwind tour of all of the outstanding work that's come out of your lab over the last decade and a half. Okay, so just a minute or two for questions. We have one from Mark Williams in the chat. Don, can you apply chemotactic cytokines and observe lymphocyte migration out from the LN chip?
We haven't done that because we don't have an endothelium in that chip, because we get sort of a host graft response. And we're trying to now integrate to get endothelium that are either from the same patient or may have decreased MHC to try to get around that. But we've done that in a bone marrow chip where we can get basically immune cells that are differentiated migrating out in response to stimuli. So it should be possible in the future.
Thank you. Okay, any other clarifying questions for Don before we move into our panel discussion? I'm very impressed with everyone that they've managed to keep to time very effectively so far today. Okay, so I'm going to ask all of our speakers to please go ahead and join me in turning your cameras on. And we'll try and get great. And then I'm going to turn it over to Amber Daniel, who many of you will know by name, if not by face. She's absolutely outstanding toxicologist that works as part of the NICEDM group, and she is the main support person for the MPS Core Work Group, and she will be moderating our panel today. So Amber, I'm going to turn it over to you.
Thank you, Nicole. It's my privilege to coordinate the MPS Core and all their activities. I'm really excited to be moderating the discussion today. So I just really quickly wanted to cover just a couple of notes for the panel discussion. We're going to focus today on the more scientific aspects of MPS models for infectious disease, and then tomorrow's discussion will have more of an operational kind of regulatory focus. So I hope everybody can come back and join us for that tomorrow. We have a set of questions that we're going to ask that's kind of help structure the discussion, and I'll ask the panelists to kind of kick off the discussion of each topic, but we do invite and encourage all the attendees to contribute your thoughts. And the way that'll work is you'll need to use the raise hand function in your Zoom toolbar to let us know you'd like to speak. It's under the reactions icon, and then when we're ready for your comment, and we'll try to go in order, I'll invite you to unmute yourself, and you should see a prompt pop up on your screen that'll say that. And of course, if you change your mind or we run out of time and have to move on to the next topic, of course, you can lower your hand to remove yourself from the queue. So without further ado, our first topic is around the critical elements of MPS systems in terms of being considered fit for purpose for studying immunological responses to disease and treatments. And more specifically, what are some major scientific and technical challenges that must be overcome to promote a more widespread use of MPS for studying immunological responses to disease and treatments? What are some best approaches for identifying and measuring infection and immune changes in those MPS models? And then what are some best approaches to study immune cells that can be used with current MPS technology? And Gwen, if you don't mind getting us started on that, please.
Oh, sure. Thanks. So the critical elements of MPS systems for immunological research, I think, initially, of course, physiological relevance is key. Your MPS system should accurately mimic physiological conditions and microenvironments of target organs and tissues, so you get reliable and translatable results. I think critical in that is also cell types and sources that are selected. It's important to select those that represent the complexity of immune systems and disease pathology, for example. Providing biomimetic cues and stimuli. So relevant cues such as, the biochemical gradients that are needed, mechanical forces, the co-culture system, so you can recreate dynamic interactions and signaling within the immune system. I also have integration of vasculature, so developing, or having vascularized MPS models that replicate the transport of immune cells, cytokines, and signaling molecules to study immune cell trafficking and more systemic immune responses. Certainly the long-term functionality of the MPS system, so you can maintain functionality of immune cells and tissues over extended periods. So you can study, chronic diseases as well. Analytical capabilities are important, so you can integrate real-time monitoring, imaging, within the MPS systems to allow for assessment of immune cell behavior and responses. So those are what I have as critical elements, and certainly there are scientific gaps and challenges as well. Capturing and the complexity and the heterogeneity of immune responses within MPS is still a challenge, especially including the interplay between different immune types and the integration of the innate and adaptive immune responses, scaling and throughput. I don't know if we want to stop and then come back to some of these points so other panelists can also weigh in. But I have standardization and validation and regulatory considerations also as key both scientific and technical challenges.
Great. Hi, Quinn. This is Dayton Pettibone. So given the complexity of the immune system, as with any tissue and incorporating it into development of a microphysiological system, most of the organs that are being adapted to microphysiological systems represent small functional units as part of the instead of the whole organ. Would this be a prudent approach to incorporating immune cells into the chips, or would it be better to have the full capabilities of the immune systems incorporated into the chips? For instance, inclusion of immune cells in native tissues, such as, in my case, testicular macrophage cells and Hofbauer cells in placental tissues. So should this be approached, first of all, looking at just the most relevant aspects of the immune system for the model instead of the whole immune system being incorporated?
Yeah, absolutely. I think we want to, to standardize the small functional units first before you approach the whole immune system. But let me let other panelists weigh in on here as well.
Can I speak, Amber?
Yeah, sure. Yes, go ahead, please.
So you've got to remember that these are fit for use systems. All right. So the idea that we're going to just build generic systems that have all this complexity and use it for everything, I don't think is realistic, because anyone who's done this type of work, it's incredibly complex the more different cell types you incorporate in, right? And so, for example, we don't have dendritic cells in our intestine chip, and we could do a lot of relevant studies. But when we wanted to do mucosal vaccination studies, we had to integrate dendritic cells into the intestine chip and link it to our lymphoid follicle chip, and it works beautifully. And so, but I don't need to have dendritic cells there all the time. So I think you really have to define what your question is. And this, I always say, it's synthetic biology at the cell, tissue, organ level, and you want to start with the simplest model you can. And if you can replicate a clinical response, you just learn you don't need all the other complexity. But if you don't, you now have a test bed to see what else needs to be added. And so, I think all the things we're talking about are right, but I just think it needs to be defined by what the purpose is.
Yeah, completely agree with that. And certainly, we can take, for example, not all of the inflammation models, just looking at neutrophil adhesion and migration, for example, you don't always need all of the cells in there to look at activated endothelial cells within inflammation. However, some of our target models where we are looking for drugs that can cause inflammation or reverse inflammation, we do include the relevant cells in co-culture and the relevant microenvironments. So completely agree with that.
Mark, you have a comment?
Yeah, I just to build on both of those things, I think it's really critical as you're adding in these cells into the system is to know that they're actually becoming or differentiating or representing the actual cells you're trying to model. I mean, putting, we put macrophages into lung systems, into the tissue, and we really have no clue whether they represent alveolar macrophages or tissue macrophages, or in Dayton's case, the testicular macrophages. There's a lot of characterization, I think, that goes into that a priori to really give yourself that confidence that you're sort of in any way of shape or form replicating tissue function.
Mm-hmm. So what about the cues, the relevant biomimetic cues and stimuli in these platforms? Certainly, those are very important. So you're eliciting the appropriate responses, and so does anyone want to weigh in on that, where we are with that and how important that is for these type of immunological research models?
Simon, I see you have your hand up. Was that something separate or?
I'd rather wait for responses to Gwen's
Okay.
question.
All right, then I think maybe Don. Then.
Yeah, interestingly, in our lymph node chip or lymphoid follicle chip, we don't get that response without flow. And the flow is in a separate channel, so it's not shear stress, and we're not sure what it is, but certainly the microfluidic, the flow is critical to get the level of immune response that we see. So that's one example where that's interesting. I showed you the example with breathing and lung viral infection, and we've integrated macrophages into that, which have a dramatic effect on the response as well. So that's adding more complexity. We haven't yet explored the interplay between those two, but I think you're going to see it in other systems too.
Right. Flow certainly is a huge component of these models, certainly because the flow, we know that, the appropriate receptors are upregulated, the transporters, all the different pieces that are necessary for complete responses can be, induced by flow. And so more static models can sometimes not have the appropriate expression levels of receptors and even tight junction proteins and other things. So flow certainly is a critical piece.
Okay, well then on that topic, there is a question in the chat that might be directed towards Dawn about, did you try different flow rates to observe the collagen alignment in their assay, and how far in terms of range collagen can align from its distance with the flow boundary layer?
In our paper, we showed collagen alignment in the lymphoid follicle chip with flow. That was part of that response. We didn't try different flow rates, but we did fluid dynamic simulations that would suggest, because of the thickness of the membrane and the size of the pores, that it's not shear stress, as I was suggesting, but we did not explore that further.
Thank you. All right, then Simon, we'll take your comment and then we'll move on to the next question.
Yes, thanks. I mean, the talks I've heard today are absolutely fascinating, and each speaker seemed to have their own source of cells, and some of them had to use primate instead of human because they couldn't get human. And when Mark was talking about BEI and the sort of support services that are available from BEI, it made me think about 30 or 40 years of experience of animal modeling, which I've specialized in, and thought 30 or 40 years ago, everybody had their own colony of mice, and it was pretty it was to some extent fairly unregulated, but today there are regulated sources of mice, rats, small animals, and non-human primates. Everybody knows exactly what they're buying from a commercial supplier. And that infrastructure that's taken 4 decades to get in place for animal modeling now needs to be in place for MPS in terms of cells and supplies of cells. For people like me who are starting out, I don't have that infrastructure available to me, so I need to tap into it, or I'll need to try to create it myself. And I really don't want to do that. I'd rather hear that BI has got the cells that I need and that everybody agrees that they're the ones that we should all be using. So an international effort uses the same sources of cells over.
Thank you very much. And we will return to very popular questions as time permits once we make it through all of our other questions. So moving on, what tissues or systems are best suited to help demonstrate and test efficacy of candidate countermeasures? Tyler, can you start us with that?
Yeah, so I think when thinking of this question, you want to start off by thinking really about how you're going to establish that infection model. So in the case of bacteria, what's the route of entry? What subsequent tissues are then going to harbor that infection downstream? And then I think from there, you can decide, like Don was speaking to, do we want to go more complex or not? If it's more simplistic, and for instance, maybe think of Francisella tularensis. after some time in infection, there's a bacterial burden that's known in the liver. So if you're looking at efficacy of an antibiotic, you might be able to get away with just looking at the efficacy of the drug at that specific tissue site. But what we're doing in our hands is kind of broadening that to more complex system with the multi-organ chip. So with that, we have the liver component, we have the kidney component, but we also then can choose 2 other organ types, that might be pertinent to that particular infection model. It's nice because you have the microfluidics that are can be vascularized, so you can simulate an IV injection. But again, if you're wanting to add something like intestine, then you can simulate, an oral dose of the drug. So all things to take into consideration, but I really think it's how simplistic you want to start out. Is that going to answer the question for you, whether that drug's efficacious or not? And then we get into, issues with the immune system and the specific immune cells that might play a role. And, beyond antibiotics, the question becomes much more complicated and more broad. But I think, with us in our hands, we're going the multi-organ on a chip route, but interested to hear feedback on that.
Mark.
Yeah, I mean, I can talk a little bit about what we think, and I would echo, basically what Don said. The fit for purpose idea is that, if you're looking for, as you said, antibiotics or direct acting antivirals, you could model the target, the site of infection. But if you're thinking about getting a drug approved or something like that, one of the first questions that you're going to get from a regulatory agency, is what's the trigger for treatment, right? And so just looking at, let's say, Francisella from a tick bite, you model a skin infection, an ulcer or a sore on the skin can then be sort of that trigger for treatment. We use, inhalational exposure to Francisella for our, non-human primate models. And in that situation, then the blood or the liver become the, some of the readouts, just like what you're saying. So, you really have to be, I think, careful and targeted in terms of what you're going to be, using to evaluate. And again, I'm really talking from the perspective of drug development, as opposed to really understanding, the disease pathophysiology.
In discussing the tissues or the systems that would best help test the efficacy of candidate countermeasures, it might also be useful to use tissues that exhibit pre-existing risk factors for susceptibility to infection of pathogens, such as the case with SARS-CoV-2 with diabetes or asthma or chronic lung disease, or sickle cell disease, for instance.
Right, and taking into consideration donor-specific differences as a whole, yes. Right? Really.
Thank you. All right, let's go to the next question, which is for Mark. A good follow-up to your presentation. What types of cell sources or other resources would help advance and/or harmonize the use of MPS for infectious diseases?
I mean, this is basically mimicking, Simon's comment, and this is something that I've really tried to think about in terms of, like he implied, what BEI does, or how is there a way that we as the government or we as an organization can provide that harmonized, well-characterized set of challenge material, well-characterized cell sources. And what stimulated me thinking about this, some time ago, I think, was seeing some of Don's early work where he took the organoids and, I think the way you described it at the time, Don, was just sort of opening them up and creating the intestinal chip based upon the organoids. It sounds like you've evolved a little bit in that sort of technology, but I mean, is an organoid bank then for, because we can obviously provide, large reproducible, amounts of organoids in a cryopreserved manner. Do people feel like that is a usable or realistic source as for well-characterized cell types that can be used to seed these systems?
Don?
If I could respond to that, absolutely. I think, in my experience, that is the best source. NIH had a huge emphasis on IPS when we started the organ chips, and you lose all the epigenetic signatures that are relevant for disease modeling, unless it's a genetic mutation. And they often are not mature when they differentiate. And whereas the organoids, are extremely great sources in every system we've tested in our experience. The one thing I'd like to raise to build on what you and Simon said is that, like, we are, what you don't want to do is to have inbred humans like you have inbred mice, right? And you want to keep the variability that you see in a clinical trial, and yet you want to know information about the individuals. And so we buy a lot of our cells. And for example, we're doing cervix chips, and we're trying to do different ethnic groups. And we've got, 2 Hispanic, 2 Afro-American, 2 Caucasian, but there's no information about these people. It's minimal. You have sex and age, and that is due to NIH guidelines about what you can give out in terms of information. But that information will be invaluable for us, right? And so I think organoid banks that are qualified, because we're all going to have to come up with qualifications in the future that are common. I think, as Simon's point is right on, but not so. We don't want to use one organoid source of intestine. We want to have that variability. But I think organoids are the best that I've seen.
Well, that's good to hear, because I that seemed to me a way to have cryopreserved rather than people going and buying up the lots of a particular lot of cell type, from the pharmaceutical companies that provide these cells. Would those organoids then be, I don't want to say a long-lived, let's say a long-lived source of cells.
Yeah, they're intellectual property issues, though, that it's controlled by one group. So that needs to be worked out. But and I will say that from the beginning, we take organoids, we expand them, and then we break them up either into small aggregates or even sometimes single cells, but small aggregates work the best. And then we plate them that way onto chips.
Nicole, you have?
I'm just interested. I want to dig in just a little bit into something you said, Don, about sort of the limitations of the iPSCs, and whether or not you think that those are really rate limiting or factors or stumbling blocks that can't be overcome, because I've seen some of the more modern differentiation protocols that claim to be able to produce like very homogeneous, very stable populations of mature hepatocytes or cardiomyocytes, or these cells that are more stable and are more mature. And then if you have, patient-derived iPSCs, I understand the loss of the epigenetic component, but you still have the genetic component to represent that variability. And again, my limited understanding here is that when you're using patient-derived iPSCs, you still have some replication of that disease state, potentially, if you're getting them from diseased patients. So I wondered if you could just expand a little bit on whether or not you think those limitations are something that can be overcome, or we should just pivot our focus to primaries and keep it there.
I'm not the world's expert on them because I haven't used them enough, and I think it'd be great to see some sort of study that compares them. But my impression is that the neuronal differentiation is probably the most sophisticated, and there you're looking for cell type specific function that could be really useful for that. And I maybe some of these others have advanced past where they were a few years ago, but in general, everyone in the field that I've heard speak about it admits that in general, they're kind of neonatal level differentiation and often not fully mature. The other thing is that we developed a human blood-brain barrier chip that had a fantastic barrier using iPS-derived endothelium that we were convinced by the field that were the brain microvascular endothelium. And we were only later did we learn actually they're not. They're actually brain neuroectoderm. And you know, so that you can often spend a lot of time and then you realize it's not exactly what you think. But my biggest concern is this epigenetic side, and I haven't seen anybody clearly analyze this, but the whole concept of IPS is that you kind of reverse back to an early state, right? And it's hard to believe you retain the epigenetic signatures that we see in disease. You know, I remember when we first did our airway chips and we did chronic obstructive pulmonary disease derived cells. That's an inflammatory disease, right? You know, you're thinking immune cell. This is not, you know, a genetic single point mutation, whatever. It retains the phenotype without any immune cells there, right? And so I'd like to see someone show me that IPS retains that same phenotype, and the same with, you know, many other conditions. It's possible, you know, and maybe there's some protocols in IPS that are more sophisticated that'll allow you to do that. So I wouldn't give up on it, but I think you really got to be sure it works. Whereas every time I've seen organoids, you know, and you know, you do the qualification that, you know, they have stem cells and they differentiate into and show many functionalities. They're usually mature and they're usually quite robust.
Thank you.
Hello. Hi. So this is H speaking. So now maybe we are talking about discussing about the single organ MPS, and in that case, so the epigenetic is some kind of trouble. But when we think about the multi-organ MPS, how about the isogenic organ? Each isogenic organs is important or not? I mean that when we use the IPS and differentiate into several organs and build up the MPS with those different organs, but those organs have the same genetic background. Do we need such kind of single background MPS, or if we construct the multi-organ system, is it okay to have different genetic background?
So you're saying if you're just developing healthy chips, not a disease model, the IPS may also be useful there, but you have to qualify that they really differentiate into mature cells. The power of being able to get multiple MPS from the same individual, that is a huge advantage of IPS. So it probably should continue to be explored in a big way. Then you're asking if you take IPS from different individuals and put them into like a body on chips. Were you asking about that as well? I missed that part.
So yeah, my question is that the first one. So put the several different tissues from one donor single IPS.
I think if you confirm that they fully differentiate into mature cells, then I think, you know, that is a place where IPS has a major advantage, and it's hard to do it with organoids. My comment was more on the disease modeling side of this.
Yeah, so Tyler, Dayton, where you're doing multi-organ, I mean, Professor Ishida, you also are doing some multi-organ. Are you using from the same donor, or is it you know, just catch as catch can?
Yeah, so we are not, but I know Tissues is working towards that. They have a XX and an XY chip, I think they call it right now, that's in development, that aims to, you know, derive from a single donor up to, I believe, 13 different organs onto one chip. So that's kind of where they're going. But at this juncture, yeah, we're pretty much subjected to using, you know, a variety of different donors or, you know, different sources. We use Fuji a lot for cardiomyocytes and supporting cell types to make our organoids. So yeah, I mean, that's what it ends up being is multiple cell types, multiple donors.
So in my non-human primate model, I've been using a I've been using tissues from a single non-human primate, though I have other tissues available. And also the neurospheres we are using in our model are a cell line that were derived here at NCTR, which is capable of neural differentiation. So while my supplies last of the testicular tissues for this individual, I'll continue to use those tissues, but of course that's a finite resource, which is eventually going to be exhausted. And I'll have to begin using tissues from another individual.
So to cycle back to Professor Ishida's original question, do we think we should use the same sources for each of these organs when they're linked together? I mean, I'm assuming that was your question, sort of, you know, global question.
Certainly should be explored to see what the difference is, right? No.
In the perfect world, I think it would be nice, but
yeah, that's what I was just going to say.
But it's not
Perfect
practical.
Yeah.
Yeah, certainly with primary cells in general, you know, it's hard to get multiple donors. And you know, I think about the primary cell sourcing we do for our brain models, the blood-brain barrier, and we tend to have to test every lot for its ability to withstand flow and shear force and be able to form complete lumens. We know we need adult cells, and certainly they vary depending on the donor. And so it does seem like, you know, doing a study with single donors might be risky because they can have very different responses. But you know, how do you standardize on cells for particular models? With iPSC derived cells, you know, there's in addition to the phenotype drift, you know, there's also technical challenges of them, you know, surviving cryopreservation, for example. Viability is an issue. So the question of cells and sources of cells, I think, is a very important one for standardizing.
Realize even organoids, when you develop organoids, which we do ourselves too sometimes, you pick the ones that grow better. So there's always qualification issue, but that is one of the criteria that you need to come up with. But you know, I think there's you know, the FDA is qualification is all they talk about, and I think we're going to have to come up with, the whole field has to come up with guidelines. I think Simon's comment about that this was done for animals is an interesting one, because maybe one can go back to see how they approach that and use something similar for the industry. I think that's a great idea.
So, for example, FDA says how many different donors are safe to say something for the regulatory document. Are there any hint of the number? No. 3 or 5 different.
I think it's going to be statistically determined by, you know, sort of doing an analysis of the results and showing the robustness.
In a sense, so do we need to get the statistical significance? So do we need to do the experiment to get the finally the statistical? Yeah. So we don't know how many we need to reach that point.
I mean, it's a really good question, because we just had this paper with Emulate where we did 27 drugs and 800 liver chips with 3 donors. And believe it or not, 2 donors was 3 donors was not that much better than 2 donors. However, all of those cells had been qualified, if you like, early on, and they probably use similar qualifications. And so if you qualify the cells well, you probably get good robustness, but will that scale to patients in the end? you need to do like 27 drugs, whether we knew the results in humans and animals, we were able to show that it actually is much better than animals, 7 to 8 times better at predicting human results. But that's not going to predict necessarily all human results, because there's always idiosyncratic responses. And that's a whole other level, right? Can you get organoids from patients who responded idiosyncratically and then make chips of those and figure out what the difference is? So that we have to remember that we're dealing with the complexity of humans. And that's the way you're going to be treating people with complex populations. So we have to deal with some mess here and figure out how to handle that.
I can say for the non-human primate qualification that we did with the FDA, they expected us to come forward, with a plan for powering the studies and, based upon expected outcomes. And so just to, that follow that statistical analysis rabbit down the hole, that may be where things need to end up happening, is design some sort of robust biomarker outcome measurement, and then be able to power your studies based upon those.
All right. Thank you. I hate to interrupt such an active conversation, but for the sake of getting through all of our topics, if we can move on. The next one is for if Dayton, if you could please start us off with your thoughts on what are the best MPS models to use for non-COVID pathogens.
Sure. So with vector-associated pathogens, these are considered a major public health concern as they result in mortality, lifelong morbidity, and strains on healthcare systems in many countries. Many of these bloodborne pathogens are transmitted through insect bites. These include mosquito-borne flaviviruses such as West Nile, dengue, and Zika viruses, and all of which have been detected in the cerebrospinal fluid of infected patients with neurological pathologies. In addition to flaviviruses and other viruses, vector-borne pathogens also include bacteria that result in Lyme disease, spotted fever, or typhus, and parasitic worms or flukes, which result in malaria and sleeping sickness, that are also important and should be considered for developing MPS for disease. To date, much of the in vitro and MPS work to study vector-borne viruses has focused on viral infection characterization, pathogenesis, and countermeasure development. Much of the MPS work, much of this MPS work has focused on development of the blood-brain barrier, as well as 3D neural spheres to model neuropathogenesis. For target organs versus the site of infection, it's clinically useful to categorize viral infections by the organ system most commonly infected. And some of these include lungs, gastrointestinal tract, skin, liver, central nervous system, and the mucous membranes, some of which we heard about today, some of the research developing those models. And many of these non-COVID models will, of course, overlap with the models being used to study COVID, such as lungs and gastrointestinal models. Models to study the sites of infection for vector-borne pathogens, as well as communicable diseases, could include skin models that include circulating blood component, or mucosal models such as vaginal, rectal, or oral. MPS models that evaluate the site of pathogenic infection. Many of these are also underdeveloped, in development. Some of the key organ targets of infection also overlap with the site of infection. These include the gastrointestinal system and the lung and respiratory system, as well as the skin and mucous membranes, while other target tissues include the liver, central nervous system, and the blood-brain barrier, as well as reproductive tissues and fetal tissues to look at issues that occur during fetal development. So I guess significant effort has gone into developing the liver MPS, as we saw today, as well as the lung and other other tissues that are sites of infection. It seems that the sites of infection have not always been used as predominantly in this type of research as the sites of sites of pathogenesis in these tissues.
Thank you, Dayton. Does anyone else have any thoughts related to that? Mark.
Yeah, I can add something. I guess just what we were talking about. Fit for purpose in terms of the sites of infection versus the sites of pathogenesis. I mean, for our group, we're evaluating therapeutics, but also vaccines. So if you're talking about evaluating the efficacy of a vaccine, if you can hook one of Don's lymph node chips up to a skin chip or something like that, can you get circulating lymphocytes, or even you could even take antibodies and infuse them into these, and that from a vaccinated subject. That would be one way that you would want to design your system to test the efficacy or of a vaccine in that setting. I find myself finding more and more away from sites of infection, at least for drug development, as to the tissues of pathogenesis.
I think our finding with Lyme disease really, like, we were really amazed right off the bat, seeing things replicated that you see in patients. And I just, following up on the last comments, that integrating complex immune function, not just circulating immune cells, into disease, into infection models, I think is going to be critical, because we're already, we collect effluents and do proteomics and metabolomics, and we're seeing things come out of these chips that we think are not only relevant for T cell suppression, but for joint inflammation and breakdown and all sorts of things. And I think that's where it's going to get interesting. And we have linked, like I mentioned, this lymphoid follicle chip to the intestine chip and to lung chips. And for example, can you infect with influenza and get antibodies produced that block infection if you vaccinate? So I think that's when it will get interesting. But I don't think the initial site of infection may be it's part of it, but may not be as critical as the target sites of organs and the immune system integrated together.
All right, thank you so much. Next question is, do we need animal MPS models for zoonotic type 1 health type pathogens? And that is for Seichi to please get us started.
Yeah, so the other day, I joined the IQMPS affiliate on the FDA webinar. And in that webinar, there are also the discussion about the animal model MPS. But in that case, so they are discussing about the production of new drugs. In that sense, so if we need to, in that sense, we need to evaluate the efficacy or the safety of new drugs in human. And the human model of MPS is more important than animal model. For example, if we can skip the animal test in preclinical, and instead of that, we have the good MPS human model, it's better it's better alternative to animal model or the animal experiment. But when today I heard about the infectious disease model, I think that for the evaluation of the virus diseases, we need to the host animal model. And in that sense, I think we need to develop the animal MPS model. And so, in my sense, it's also a sort of fit of purpose point. So, for the drug development, human model is better and required, but in case of the virus experiment, animal model is also required. That is my today's impression. Yes.
Yeah, I'd like to comment on that. I also participated in the IQ MPS workshop, and there is interest from a lot of people within FDA to develop animal models, MPS, to compare to the historical data or concurrent data in vivo and in animals, and to compare with the data from human MPS and clinical data to gain confidence in the human MPS for regulatory use, because it's usually not practical or ethical, to possible to evaluate the outcomes of a drug or some therapeutic in humans. So the animal MPS will provide a means to bridge that gap by comparing data between the animal MPS and human MPS.
To follow up on that, FDA has funded us to develop non-human primate lung chips. With Mark Williams' help, we've actually done that and done viral infection models, and they look very similar to human with, let's say, influenza, but they're also distinct. And I will be pulling this together, I think, in a publication soon. But FDA, exactly like you just said, they explicitly wanted to help decrease the barrier for the pharmaceutical industry to use human chips by being able to compare non-human primate chips to non-human primates and then having human chips. We also done mouse, rat, and dog in the past. We've done rat and dog liver chips, compared them to human chips, and shown that we could mimic species-specific liver toxicities, again, to help the pharmaceutical industry compare apples to apples. But in the end, I think the most powerful part of animal chips is One Health, the idea of modeling, animals as hosts for different infections that are coming down the pike. And I do hope we see funding for that in the future, which has not been around in the past, I would say, hopefully after COVID.
Gabby?
Hey, Amber, can you hear me well?
Yes.
Okay, awesome. Yeah, I just kind of wanted to sort of second that and sort of give maybe a more a philosophy approach to One Health, because I think oftentimes we use animal models sort of to improve animal, not so much animal health, but human health, and test, different interventions and MCMs. So a lot of the animal chips or animal MPS is actually seen within the perspective of how can human health be improved. But I think when it comes to One Health and kind of looking at zoonotic diseases, which can affect both human and animals, I think the MPS field gives a very unique opportunity to use a reverse translational approach. So let's say if we have a human chip or a human airway chip, how can we maybe adapt a protocol to have, a porcine lung chip or something else that could be easily adapted and then sort of help advance the veterinary field? Because I think there's a big, as Dr. Ingber mentioned, there is a big discrepancy in the funding. So there's a lot of funding available for the human side of things, and the veterinary side really struggles with development. And I think MPS hopefully will help that in the future.
So in that sense, so I'm curious about the cost of the experiment. So for the Mediterranean world, maybe you cannot spend a lot of money like the drug development. In that sense, do we need to reduce the cost of the experiment?
Yeah, I think that's a very good point. So I think just trying, or when performing experiments, let's say on a zoonotic virus or a bacterium, maybe incorporating sort of an animal chip as a control or as an expansion of a human experiment might be an avenue, or as Dr. Ingber mentioned, having maybe different avenues of funding in the future might help really in the future, yeah.
Yeah, sorry. So, and also in that case, so maybe the more simple method is better than the complex one because of the cost. And in case of humans, so maybe cost is not so much problem, but in animal case, maybe the simple MPS is better than more sophisticated, but physiological one.
I think in the perfect world, the more simple systems would be great, but I'm not familiar with all the steps that are required for approval of drugs to be used in the veterinary care world. But I can't imagine they're much simpler or less, costly than what's used for humans. So I don't know. I think there's still going to be that level of rigor and data-driven packages still required. So it would be nice, but I don't think that's likely. But I may be wrong.
All right.
Sorry. So in the case of the IQ-FDA discussion, so it's just the bridging of the in vivo and in vitro data by using the animal in vivo, in vitro data. But when we finish in the future, when we finish the comparison and have the confidence that the MPS reflects the in vivo situation, so do we still need the animal model, or just the human model is enough to develop the drug? Maybe not in the virus case, but simply the pharmaceutical purpose. Is that the ideal goal of the human MPS?
That may be a probable scenario if there's enough confidence in the human models. Then the use of the animal models might be a shorter term use of those, or have a minimized usage in development of drugs.
Yeah, I think I would, that's sort of what the party line, right? Is you're never going to be able to get rid of the animal testing, but maybe down the line. We thought a lot about animal chips because a lot of the pathogens that we study are biodefense pathogens, and their approval then has to go through the animal rule, where there's no possibility of having human data. And so that was a situation where the animal chips would then be bridged actually to the animal data to try to, utilize those MPS systems to mimic or provide what animal data we were providing to FDA to support, drug approvals for those sorts of diseases.
All right, thank you all so much. For the interest of time, we've got a couple questions left, so we're gonna move on. So next question is for Jeremy to please start us. And that is, what do you think are seen as being the main advantages of exploiting MPS for infectious disease research?
Yeah, I think that one of the big advantages we can see in or foresee in microphysiological systems is that can we better predict? Obviously, we're trying to better predict what's going to happen in actual human beings instead of the animal models. And so I like to think, we're using the animal models to ultimately, in my realm, I'm trying to minimize human suffering, right? And so where can I use a tool such as microphysiological systems to avoid some of the more particularly painful regulatory hurdles to bring a solution or a drug or a treatment to market? And I think that brings us back to the question of what specifically is the problem we're trying to solve. And so I think there needs to be a lot of specialty here. And when I think about this in the context of respiratory disease and disease transmission, humans are an exceptionally diverse species. And when we look at infectious diseases, they do not present similar risks across a population. Sex, genetics, behavior, including like long-term behaviors, which could predispose any individual, your immunological profile or other risk factors, they can influence that probability that any one individual will become infected. So I think the microphysiological systems have a lot of promise and that we can work towards developing. And I'm thinking more big picture here, right? These are some of the problems that are not immediately to us, but something that I think a lot of the people on this workshop today are thinking about is how can we establish those populations? And when I say population, banks of cells of specific at-risk populations, be it a smoker, be it a diabetic, because when you look at some of these respiratory diseases, when I think of what is transmitted and where do we see the most pathology is in some of our people who are the least fit, right? So when you think of the data that I presented today, and I showed you how the aerodynamic particle size shifted the ID50 or the median infectious dose in a SARS model. And so that ID50, like we all look at it, ID50, LD50, but really I think one of the most important things that we often miss in animal models, and part of that is because we're always looking at the 3 Rs, is that ID10, that ID1. Like really, when you look at a pandemic, that ID1, that ID10, like what is the amount of any infectious agent that is going to infect 1%, 10% of the problem? That's still an enormous problem. 50% is just a tool, as we all know, that we use for estimation to compare the relative potency of various agents and strains and variants across the board. But the thing that really needs addressed is really the most sensitive population, because those are the most at-risk populations. Another thing that I think we may be able to consider for microphysiological systems, at least from an aerosol standpoint, or this could really be in any local treatment type of application, is that when we consider respiratory therapies that are being developed for things like tuberculosis or early stage respiratory infections, when you might have an inhalable, and these are used in COPD, when you might have an inhalable test article or drug, sometimes those are difficult to administer based on either the patient or sometimes they're specifically delivered via respiratory therapy due to systemic toxicology issues. So I see microphysiological systems as we start getting ever closer to developing the ability to repeatedly deliver aerosols to the air liquid interface and a lung on a chip system. We would have an ability to get around that enormous hurdle of early in the drug development process of synthesizing or obtaining enough of your drug or test article to test in animal models, which usually need gram quantities, if not many gram quantities to test. And we can get down to possibly screening promising compounds when we are still at maybe the milligram phase. And I know that's a big amount to people who are synthesizing these things, but even at the milligram scale of new drug development, could we use those new drugs in a lung on a chip system to see if there's any promise there as far as testing, infected cells or resistance to infection with certain cell types? So I think there are a lot of different areas there that we can establish microphysiological systems and infectious disease research. But I do think it is going to take a lot of investing in establishing those diverse, highly susceptible populations to answer some of the most important questions. Right now, we're getting them, the core methods established. But when I look at the problems I really want to solve is if I can prevent disease in that 1%, can I influence or greatly mitigate the probability that an infection becomes a pandemic based on my ability to prevent that most probability or the most likely population to become infected, prevent them from transmitting the disease as they might normally. So those are kind of my ideas on this. Oh, and again, apologies, I don't have a camera. We're not allowed cameras here. So, and I'll open it up if anyone else has any other ideas.
One thing I just add is that we've not mentioned, and I usually do in my talk, but I didn't have time, is I think another real power of the microfluidic systems is the ability to model drug pharmacokinetics, like drug exposure profiles that actually are seen in patients. I think we waste a lot of times just bathing cells and culture and drugs, and then when you get to patients, you can never get a meaningful dose. And I think being able to incorporate that early on fits in with using low amounts of drug with clinically relevant doses could really accelerate things.
Yeah, I absolutely agree. And I've spent a fair amount of my career looking through those different phases where we'll take a drug and we'll run it through PK before moving it into an animal model. And yeah, sometimes it doesn't get out of the PK, or if it does, it doesn't make it through the clinic. So absolutely. Before, when you think about those, what are some of the things that we invest too much money on for dead drugs? Some of it could be the wrong kind of PK study, right? Or the wrong kind of efficacy study. So when you scale it down so much and you look at something like, for example, the emulate chip where you need like 60 microliters is the volume in one of the chambers. So the scale and the concentration of drugs that you need to even measure the rate that it's being metabolized or cleared or whatever your assay is, it's a completely different scale. So I think it opens up a lot of avenues where we can answer some questions that we've been relying on animals for. But once we spend the money to get to the animals, we're so invested in the drug, we might not be fairly judging it. And at the same time, we could have screened an additional 50 drugs, but instead we've been synthesizing a boatload of our favorite.
Mark?
Yeah, just I mentioned this, and actually Dayton talked about it in his presentation as well, is in the infectious disease world, we struggle to find the right models where the preclinical models, whether it be mouse, whether it be rat, NHPs, are susceptible to infection by that particular pathogen. And so I think, that's, for SARS, we had to create a mouse adapted, we made, human ACE2 transgenic mice, these sorts of modifications. I apologize. Somebody talked about the interferon. Maybe it was you, Dayton. The interferon deficient mice. Those are a really common model in the infectious disease field. And the question is, are these relevant to evaluating efficacy, evaluating disease? And so I think having the MPS with the appropriate species, Dayton has got the rhesus, whether you got humans, or potentially with the different, backgrounds like what Jeremy's talking about, gives you that a little bit more power of being able to understand I don't want to say normal, but normal, quote unquote, disease processes and responses.
All right, thank you so much. We're going to move on to our last planned question, which is for Dawn. Considering the pace of development of MPS, how do end users navigate the sea of models? to identify those that are best suited for their research needs.
So, we're a little over time. So just really briefly, I mean, I would say, there are organ chips that are higher throughput, that lack certain features like mechanical cues, and others like the ones we work on that are lower throughput give you more complexity. And I think you need to choose how to approach the question that interests you. I tend to think of our chips as really replacing animal studies, where some of the other systems and organoids are a much better high throughput type of system that can get you to the point where you could kind of ask questions that we were just talking about, with the right drug exposure profiles, with the complexity of the physical environment. And so, there are questions where you want to have multiple organs, and some of the systems you heard about, like Tissues, is really already set up quite nicely for that. We can link hours for that purpose, but it requires a lot more labor and it's a lot more complex. So, I mean, I think you got to look at what's out there and realize that all these commercial products are evolving over time. They're very expensive now, but they're decreasing costs with economies of scale. So all of this may change over time, because some of it's based on cost, obviously. But I think, you have to think about the fit for purpose question and make sure that system fits your purpose. So I think I'll end there, because I know I have to run off.
I just wanted to add one other thing to that, because if we're talking about using different systems, it becomes then, what is the data package that you're taking out of that? What are the markers of the cells? What are the gene expression profiles? I don't know what it is, but that you're going to bring forward, since we're talking about putting together regulatory packages. How is the FDA, or how is whatever regulatory agency going to want to see data to compare Tissues versus Emulate versus organoid versus whatever systems, to be able to gain confidence in the data that are coming out.
So I should respond to that, because this is a very good point, that in my own lab we buy cells and qualify them internally, and they're often different sources. But Emulate and other companies need to qualify their systems, and that's what Emulate did with their liver chip, for example. They sell you the cells, they sell you the medium, and they did that so they can do what you're just asking for. That's why they published the paper. That's data they will share with anybody who wants to use it for a regulatory package submission. I think all these companies need to do that, because they may not be the best cell that each company identifies, but as long as they qualify it, it's qualified. And there's no best animal model, but they're qualified. And so I think that is where the field needs to go. And I think that's why academic lab MPS are going to help advance the field, but they're not going to be used for, to move a drug to the FDA. You need companies to do that.
Yeah, completely agree. I think it's going to be critical for that at that level.
All right. Well, thank you so much to all of our panelists. I think we'll try to end there since we are over time, but thank you all for contributing, for joining us today, and for a really great and interesting and I think robust discussion session, so much so that we've gone almost to the entirety of the end and left little time for Nicole, but I'll go ahead and turn it over to Nicole.
I don't need but 2 minutes just to say that this was just an incredible half day, really, really exciting work presented, and very engaging panel discussion. So I just want to say thank you so much to all of our speakers for your contributions scientifically and your thoughtful input into the panel discussion. We have an equally exciting day ahead of us tomorrow. So please remember to join at 8:30 a.m. on the Eastern time tomorrow. We're going to be hearing from our colleagues at NIAID, at various companies, Draper, Matech, CN Bio, Quadram Institute Biosciences. Simon's going to be talking to us from the perspective of the UK Health Security Agency. So, and then we'll have another panel discussion at the end. So really looking forward to tomorrow. Thought today was absolutely outstanding. Thank you all so much, and we'll see you tomorrow. Enjoy the rest of your day or evening, wherever you are.